Welcome, good evening to this Innovators Conclave. So today you are going to be witnessing a galaxy of innovators. And the best part about this webinar is that it's a mixture of ophthalmologists, doctors, physicians, scientists, and even industry leaders. So it's got a mixed bag. And all of us are going to play the role of different leaders at the same time, specialists at the same time, learners from each other. Now, talking about roles, we kept the theme as acting, like an actors guild. And as you all probably have seen the creative before logging in, we've got two keynote veterans who are going to give us the keynote addresses. They are there because one of them has sort of led Indian ophthalmology for a very long time, both in this country and elsewhere. And the other one is actually an industry leader who has made a mark for Indian industry, Indian ophthalmic industry, not just in our country, but over the world. And then we're going to go on to established inventors, developers, discoverers who've sort of either patented their products, published extensively on it, or even created products and even companies. So again, there you have a mix of doctors and scientists and industry people. And then finally, we're going to have the debutants. These are by no means young achievers. They are actually super achievers. It's just that their ideas are now growing from their mind into presentations and they want to take it to the next level. So we are here today to learn with you about what innovation is in ophthalmology in our country. And your questions have already started pouring in. Thank you for that. We'll try our best to accommodate as many questions as possible. What we've planned here is that we have an eminent list of panelists. So before that, let me welcome and introduce my co-moderator for this evening, Dr. Chaitra Jaydev, a friend and colleague from Narayan Nethralaya. And Chaitra?
Hi, everybody, and welcome to this meeting, which is even beyond academics. It's going to be a lot of research and a lot of innovation, which our field is now, I think, very, very famous for. And ophthalmology is, I think, literally the stepping stone now to a whole lot of research, and India is a big figure in all of it. So we'll start off today's program. I think all of you all have already seen it. Anand?
Yeah, so let's begin by actually introducing our panel. The speakers will anyway get introduced as we go down the list. So to start off, I'd really like to introduce Dr. Tara Prasad Das, somebody who does not need an introduction, a Padma Shri, currently the vice chair of LVP. He's been a sort of a senior mentor to so many, almost generations of ophthalmologists. And although I have not been his direct student, he's been a mentor to me as well and to scores of other ophthalmologists. So, sir, we welcome you.
Chaitra? I couldn't agree more, Anand, and we all look up to TPD sir, all of us, and especially in retina, he's a figure to look up to. Also, great pleasure to introduce to all of you all Milind, a dear friend. He's also very, very popular. He's a consultant ophthalmologist at LVP and adjunct professor of ophthalmology at the University of Rochester, New York. He's completed his fellowship in ocular plastic surgery and facial aesthetics from University of California, LA, and he has almost 18 years of experience in eye plastic surgeries and over 200 peer-reviewed publications. Nice to have you here, Milind.
Our next panelist is yet to join us, but I'll introduce him all the same. Dr. M.S. Ravindra. He's a sort of father figure for ophthalmology, not just in Bangalore and Karnataka, but across the country. He's the founder and director of Karthik Nethralaya here in Bangalore and has served as the past president of the Karnataka Ophthalmic Society and a very active member of the state at the All India Ophthalmology level. We'll welcome him again when he comes, but that's Dr. M.S. Ravindra. Chaitra?
And now I have a very young ophthalmologist, and that is Divakant. I think all of you all again know him. He's a vitreoretina consultant at IQ Super Speciality Eye Hospital in Lucknow. He's also the General Secretary of Young Ophthalmologist Society of India, YOSI, which is extremely an active group, and the editor of the Young Ophthalmologist Times, and they come out with extremely good publications. Welcome, Divakant.
Our final panelist for this evening is Dr. Naren Shetty. He's the vice chairman of Narayan Nethralaya. Again, young, energetic, full of ideas, and is a leader. He's led all of us into innovating research and taking us to the next level. and it was just befitting that we call him as panelists. So those were our panelists, and I think let's begin directly now with our speakers. So we'll introduce them as and when they speak. We have two veterans, as I mentioned. This team is going to be all the acting guild. So our first speaker, again, needs no introduction. Dr. Lingam Gopal sir, almost synonymous with several of his discoveries and findings in ophthalmology, led Shankar Nethralaya as director, and is currently a senior consultant and associate professor in the NUH hospital in Singapore. So he's actually addressing us from another time zone. Thank you, sir. Thank you for consenting to be our keynote speaker. And sir is going to talk about how India can actually lead ophthalmic innovation. He'll probably speak from his experience. And as we can see, it takes not just from this country, but his experience all across the world. Sir, over to you.
Let me share the screen first.
As sir gets on his screen, the audience is, I think, joined us from Facebook, YouTube, and the Entod link. Please keep your questions going. We are collating them, and we will address each speaker at the end of their talk with your questions, as much as we can.
So you're able to see the screen now?
We can see you, sir, and we can hear you. Please start.
Thank you. At the outset, I would like to thank Dr. Anand and his team for having included me in this. How can India lead in ophthalmic innovations while I'm actually talking from Singapore? That's okay, because I did spend a lot of my life in India. So I think I'm eligible to talk on Indian innovations, even if I'm in Singapore at this point of time. I felt it's a rather difficult subject to comprehend and put it across in 12 minutes, but I try to do my best. First, I thought to introduce you to the subject of innovations in ophthalmology. What are really innovations? They could be in the field of surgical techniques, in the field of medical therapy, which could mean not just drug therapy, but gene therapy, etc., etc. Innovations in the new tools for examination, innovations in new surgical tools, innovations in applications of AI in ophthalmology. I'm not going to talk about all those details because that will be beyond the scope of a presentation. But I thought it's important we should know what forms the breeding ground for innovations. We require an input from several sources before it can start producing innovations which can have an impact on the society at large. One is identifying an unmet need, a perceived inadequacy of the existing solutions, an alert mind which is able to identify these unmet needs. Of course, you require support from peers and institutions. There should be technology to support your innovation program. There should be availability of expertise in allied specialties, which is very important these days because most of our efforts will be collaborative in nature. And you require financial support for research and infrastructure to support the quality research. So briefly on identifying the unmet needs. These could be random source ideas generated from random thoughts or from while we teach residents and fellows who generate some ideas as you discuss with them, or from discussions among focus groups. I again wanted to specify that meetings targeted to identify unmet needs produce a lot more identified problems that can be solved rather than diffuse meetings like a retinoblastoma meeting or an oncology meeting. It becomes a general meeting not focused on unmet needs to be identified. So if you want to do quality research, you really have to have focused meeting to identify unmet needs. What about availability of technology in core field and allied core fields and allied fields? As I referred to you in the first few minutes of my talk, cross-specialty collaboration is a must for innovations. There is no way a single ophthalmologist working within his own environment can produce any reasonable innovation that can have an impact on the society at large. India does have expertise in most specialties, but the caveats are very often the driving force is only from one specialty, not from the other. And ophthalmologists may be interested in doing some innovation, say in developing some instrument or a tool, but from the other side, say from the technological institute, there may not be equal interest and equal driving force. What is required is to bridge this gap. There's also issue of funding for multiple specialties, especially from the grant agencies. Trying to apply for 2-3 different specialties is not easy. There's also need for synchronization of the time, manpower, planning, etc., to produce any kind of good quality research. What about financial support? In terms of innovations, innovation that need no investment at all, or like tweaking a surgical technique or a new use of existing techniques or drugs or devices which are already existing, they don't need an investment. Investments are innovation that need a little investment, but something any hospital or institution of repute is able to set aside is something which can be in-house research. There can be a little more money generated from the grant agencies, which can support some better quality research by supporting manpower and few consumables. But if you are really looking at something that has a major impact on society, you are looking at big ticket innovations, not innovation that can be solved by these 3 steps. So the innovations that make a major impact, they come from identifying first an unmet need, for which, as I said, you need a focused meeting which focuses on identifying unmet need, not from a generalized meeting. You require a structured approach. Once you identify an unmet need, you have a structured approach to identify a solution. That means you identify first who are the people who can be the key players in trying to generate the solution. It may be a microbiologist, a molecular biologist, a biochemist, an information specialist, or a technician, something. So all these you need to identify from the unmet need. Once you identify a structured approach, then you try to identify the people who can support you in this quality research and bring them together. And then as a group, you try to produce a document which can act as a beginning point. And that is the way a real quality, high quality impact generating innovation can be made, not by one man sitting and getting a random idea and trying to convert it into a major innovation. A good grant support is required till the product is brought to a stage of commercialization. Very often we start off well, but we just drop out halfway through because of lack of good financial support. So what India has, it has quality clinical ophthalmic services both in private and public sector. Once upon a time, people used to believe that the public sector is lacking in quality clinical ophthalmic services. It is no longer true. Excellent ophthalmic services are being provided by public sector as well. There are quality clinicians who are interested in innovations. There's no doubt about this fact, by the very fact that there are so many people who are collected for this particular webinar. There is some support now for low-cost ophthalmic research, and there's some interest from industry for low-risk investments. That is what needs to be changed. And what India needs is a top-down support for quality research. The financial support for research should be in a way that supports high-impact research, not just supporting you, giving you 1.5 lakhs in a year. It goes no way in producing a major innovation. Inter-institutional collaboration should be supported at all levels. And some degree of support for instruments, which is lacking in most of the grants that we get from the government, should be available, not just manpower and consumables. There should also be a change in the mindset of people. The researcher's goal should be long-term, not at publishing one paper and forgetting about it. It's about taking it all the way to commercialization and making it useful to the society at large. And also the knowledge of concepts of startup companies, incubators and accelerators, which are important to convert an idea into a product. It's not enough to be an ophthalmologist, but you should get an idea as to how to approach the right people to be able to convert an idea into a product. I think beyond this, I may not be able to talk much about how India can take a lead in ophthalmic innovation. So in summary, innovations are a way of life. India has tremendous need as well as potential for innovations. We have all the elements necessary, both minor and major, in our country to have major and minor innovations. But what is needed is a chemistry, the driving force, and the momentum to galvanize these into a global force to reckon with. Thank you very much for your attention.
Thank you very much, sir. That was like a very comprehensive checklist, right from how an ideation, a special focus group, meeting the right collaborators, what strengths we have, and what we need to make that go forward. That is really well put. While Chaitra is collating the audience questions, we have a question for the panelists. So I'm not, anyone from the panel can take it. We probably start with TP sir, because he's probably had experience working from an organizational level. The question for you is, he's talking about finding the right collaborators. AIOS, which is our parent body, has a research wing. So how do you see AIOS's research wing helping in sort of fostering this research environment and helping for research opportunities for people who probably don't have ready access for the bigger facilities? We can start off with Dr. TP Das, then we'll go on to Dr. Suvin, and then we can actually ask Dr. LG sir again. Dr. Kim is also here, so him also. These three for this question.
So Dr. TP Das has to unmute his mic.
Sir?
I'm sorry. AI voice can actually play an important role in fostering research in young people. But currently, AI voice is more interested in doing something else, and which I thought research-wide Lingam talked about. It not necessarily has to be a product. It also could be idea. I've always thought that, like DRCR Net has done a wonderful clinical research, because to collect 20,000 people together is not easy, but then you have a huge opportunity to do clinical research in all specialties, not necessarily only diabetes like DRCR, but then any area which you can work. That will be a huge contribution to research methodology and research ideas in the country, which AIOS can do. On the other hand, organizations like Nara Nethralaya to Arvind to Shankar Nethralaya to N.V. Prasad, all these guys can join hands together as a private players and then join hands with the AIMS and PGI, GIPMAN, like that, a public-private partnership, then join the industry to make it a real incubation hub, which has not happened yet. Look at the story of how India improved a CSR cataract surgical rate in the 1990s. It's just not because to use the World Bank loan to train people, but also two industries, pharma and device, particularly IOLs, etc., join hands. Because three forces, pharma industry, the device industry, and training, which we all give to trainers and trainees, put together improve the cataract surgical rate. Similarly, quality outburst of the Indian ophthalmology. The same thing can happen. I will believe that AI will someday will take a lead on a huge clinical research in all areas in ophthalmology.
Thank you, sir. Actually, ARC in the wing of the research in AIOS has been doing a lot. Some of us, in fact, on the panel and speakers have even participated under the apple tree, which fosters great ideas. And I think the next level is to take those ideas to sort of making it a commercial product, what Dr. Gopal said. And I think that's where we need that link, and that's where we need the support. We'll quickly ask Dr. Kim if he's there, sir. At Arvind, I mean, Aurolab aside, because we're going to have a talk there, Aurolab aside, how do you foster the idea of research among your faculty and sort of disruptive research, something that's really new and making it from idea to product outside Aurolab?
So you have to unmute your mic, sir.
Can you hear me now?
Yes, sir.
Yeah. What we are doing, we have been trying, but I don't think we have been successful yet, is we have created an innovation club in the organization. Some of our team members have been doing extremely well in fostering this innovation, especially which has come from some of our residents quite a few things challenges were thrown to them. We keep asking people to give problems and then ask others to give ideas on how these challenges could be addressed. This is all on our website, where we have an innovation area where people look at it and then try to see if we can work on those challenges. But still it's at the infancy. We really need to move a lot ahead, especially in this current times.
Thank you, sir. Chaitra, any audience questions? You can shoot that now.
Yes, there is one question to Dr. LG from Dr. Shantanu Ghosh in Kuwait. He says, could you please highlight the role of ophthalmic entrepreneurs and alternate career options during the post-COVID era?
I don't claim to be a specialist to give this advice, really, but I would say ophthalmic entrepreneurs is a a good term to use, I agree, but I don't understand half the time when an ophthalmologist turns into an ophthalmic entrepreneur, unless you have an idea which can really be converted into a product. That's very important. If you have an idea which can be converted into a product, and if you're willing to work with investors, etc., then you become an entrepreneur. Then, of course, you go into incubators and accelerators. There are incubators and accelerators available in India as well, and try to get into the correct group which can support your research that can be taken to its fruition. As I said, very often in an academic environment, we are not able to take it to the fruition unless you're able to link up with the correct collaborators. And there you require people with the knowledge of investments and incubators and collaborators. Only then it becomes a useful product to be commercialized. And I don't know whether it's pre-COVID, post-COVID, any difference is going to be there. I'm not sure about it. But basically, that is a approach and attitude that we need to take.
Thank you, sir. A very quick word from Dr. Yes, sir, please.
Can I add? Well, there is other alternative path Lingam and everybody has is to become a Robin Cook, write books, an ophthalmologist, or become politicians like happens in Singapore.
Good idea, sir.
I think
a quick word from Dr. Naren.
Before we go on,
please, sir, go ahead.
I had one point about the AOI's role, which I was having in my mind when I posed the question, was that big institutions like LV Prasad or Shankar Nethralaya, Narayan Nethralaya, Arvind have no problem. They have good facilities to support their research. But I'm talking about the people with small groups or individual practitioners who have a brilliant idea, but they are no way able to convert it into a probable research platform. So is there a way the AOIs can link like-minded people and tell them where they can reach out to convert their idea into a research project? That's basically where AOIs can play the role of linking people. That's what I meant.
We'll make Dr. Chaitra, who's Joint Secretary AOIs, take up this question with the Executive Board next.
Good one.
Homework for you. So a quick question to Dr. Naren Shetty before we move on. Dr. Naren, you sort of lead Narayan Nethralaya, which is a private teaching hospital, but nevertheless private, and you've sort of set up a research lab, which doesn't, at least initially did not pay dividends in terms of financial gain. So what makes you go towards that innovation, and what was the calling?
Definitely at Narayan Nethralaya, we have always followed a simple theme that it's not that we are always trying to push the institute. We grow with the people. So we let people grow and we grow with them. That is the most important theme our chairman has done, which is very, very vital. And we need to understand and not try to hold something so strong that you're going to lose it. When you let people grow, then simply the amount the institute also grows is 100 bounds. So that is one, I feel, a key a secret which Narayan Nethralaya has. No more a secret, but that is something which really everyone should let people grow, and this will make a lot of difference. Yes.
Thank you very much. Before we move on to our next keynote, I'd like to welcome our panelist, Dr. M.S. Ravindra. He had a small problem connecting, but thank you, sir, for being here. We introduced you already, and you're muted, sir, but nevertheless, we will get back to you when we are asking you a question.
Sure.
So welcome into this conclave. Our next keynote speaker. Yes, please. Go ahead.
Just a small comment about the previous question. Until the bigger bodies come into some kind of a plan, till then I feel best is to identify a mentor whoever's there close by to you, who's been successful in the same, and approach him, and you can take his guidance. That is one thing which people should do. This way, actually, you can have, you don't have to start from the scratch. You will get, you'll be automatically a few steps above. And then from there onwards, your mentor will help. So I feel identifying a mentor who's close by to you, who's been successful, is the key, I feel.
Thank you. Yeah, finding the right guru. Great.
Yes, the guru.
Our next keynote speaker is Mr. R.D. Sriram. Again, someone who is very well known in the ophthalmic industry fraternity. He's been the managing director of Aurolab for several decades now. And as we, that's really a household name for ophthalmologists, not just in our country, but all over the Asia Pacific and the rest of the world. And to take Indian ophthalmic industry to that level of excellence and get it noted and been awarded is something that he's really made our country proud. So Mr. Sriram is going to take us through that journey of Aurolab, and from that, how we can learn and get inspired at the same time. Over to you, sir.
So thank you, Dr. Anand. Thank you, Dr. Anand. I hope you are able to see the slides.
Yes, sir, we can.
We can see you, sir. Yes.
So I'll talk about the journey of Aurolab, from the context of taking the ophthalmic industry to the world. So my presentation will be basically a need for an entity like Aurolab when it was started, and how we began our journey, and then the catalytic role played by our IOL division and IOL products, and the growth of other products that we have with us. And then I'll talk about some unique products that we manufacture, and then also the global impact that this organization has made, and then I will leave it for discussion. So the need for an entity like Aurolab, basically Arvind, this was an entity that came into existence from Arvind. So Arvind treats the patients irrespective of their ability to pay. So whether the patient is able to pay or not, he gets the same kind of treatment, whether it's a free hospital or the paying hospital. So in the late 80s, when the lenses came into the market, this ethos of equity in care got compromised, because the hospital was not able to buy a lens at a very high price and then give it free. So this was a situation. This was the situation which led to the birth of Aurolab. Basically, if you look at the situation at that point in time, most of the innovations even today that happens in the West, so it is all targeted for those markets and then priced what those markets can pay. Such technologies, which are also needed for the developing countries, when they come here, only the top tier of the society is able to bear the cost. As a consequence, the market remains very small. Sometimes this technology, when they get obsolete, they don't even come to the developing countries. There are many technologies, they die in the West, and then they don't even come to the developing countries. So the intent, when they bring such technologies to the developing countries, the intent is to maximize the profit, but they're not for spreading it for many people. So there is a lack of intent in diffusing these such technologies. So that was the time when Aurolab came into the existence. So we started off with only 10 members. It was a very humble, small beginning, and the technology we bought from US. And at that time, the hospital, Arvind, was using about 30-40 lenses per day. So they decided that we'll go for about 150 lenses capacity. But today we are making about 10 to 11,000 lenses per day. So we have grown over the years. And then when we started selling at a price which our country and many other developing countries could afford. It was roughly about 270 rupees is what, when we started in 1992, we started selling. At the time, the imported lenses were in the range of about $100 to $150. So today, the IOL division makes everything from the 3-piece lenses, which we started in 1992, to the multifocal lenses. We make the whole range today. And last year, we have supplied close to 3 million lenses to more than 150 countries. We believe the number of surgeries, cataract surgeries in the globe is somewhere between 28 to 30 million. So if that numbers are correct, then we have roughly about 10% of the global share in terms of units. Apart from the lens manufacturing, we have also added capacity to make fine cartridges, which needs a very precision molding. And these cartridges doesn't work if it's not coated well. So there is altogether a new technology, a coating technology, which we have developed in-house. And then we are also making the hydrophobic material in-house. So all the key components we try to make in-house so that we can have a control on the price as well as on quality. So we were the first one to get CE certification in 1998. That was at 99. So it became mandatory in Europe in 1998. So yeah, just a year after, we got the CE certificate. So if you look at the impact that that this has made, this gave Indian ophthalmologists to do more surgeries. And because entities like Aurolab came into the market with a lower price, and also gradually increase the bar, like getting CE certificate and all of that. The quality also improved a lot. On one hand, the price got lowered, and on the other hand, the quality got improved. As a result, I think the India is able to do roughly about 6 to 7 million surgeries today. It was almost less than 100,000 surgeries in 1990. When I say 100,000, I'm talking only about the IOL surgeries. So it was 1 million surgeries. Because of the good quality outcome, the market also grew 6 to 7-fold, all within 2 decades. If you take China as an example, which has got a similar population, they are doing only about 2 to 3 million surgeries, because they use mostly lenses made in the West, like the US lenses, which are very expensive. So that market has not grown because there is no product like it is available in India in the affordable prices. So this is the impact, I think, that Aurolab has initiated, and then many companies followed, and then this all gave a good strength for the Indian ophthalmologists to accomplish a number like 7 million surgeries. Then we started in 1997 the pharmaceutical division. Basically, at that time there are a good number of pharmaceutical eye drop manufacturing companies were in India. So we focused only on those drugs which were not available, and those drugs which are called orphan drugs. The orphan drugs are drugs which are needed by a very small section of the population. So many large companies, it will be uneconomical and unviable for them to manufacture. So we targeted those drugs. So in ophthalmology, if he is not getting any drug elsewhere, so they can approach us. So those kind of drops we started manufacturing in 1997. And also there are some drugs like silicon oil, perfluorooctane, all were imported at that time into the country at a very high price. So we targeted those kind of drugs in the early times. So now we have about 9 products which are CE certified, and then we make a variety of eye drops. And then we are one of the key players in OVDs. And then, as you know, that India is a supplier for the global, India is called the pharmacy of the globe. So many drugs, generic drugs sold in the US are from India. I think very soon, it will be the case with eye drops as well. So many Indian companies can easily go to US and other markets in the years to come. So we started the suture needle division in 1998. Around this time, when the lenses price came down, it has come down to the price around in 1998. The price of the lenses were in the range of about 200, 300 rupees, whereas the suture was a bit costlier than that. So we thought, this is something to be addressed so that the cost of the cataract surgery per se can come down. So this technology was closely held technology. It was not available like the IOL technology. So we were lucky to get a partner in Germany, especially in the Swiss-German border, which is known for very, very precision engineering. So we were able to get hold of to them, and then we started manufacturing the ophthalmic sutures. But as of today, there are only 4 companies in the world that makes the ophthalmic sutures. 2 of them are in US, 1 in Japan, and we are the only one in developing countries. So because this needs a very precision needle profile, and then the demand is not great, so it offers a very unique market to us. So our goal is to become a global leader in ophthalmology sutures. I think as of now, we are the leaders in India, though Johnson & Johnson is there. As far as ophthalmology sutures are concerned, we are probably the leaders in India and also in many developing countries. And then we are a late entrant for the microsurgical blades used in ophthalmology. So we wanted to differentiate ourselves from others. So we had a very unique packing. We have cut down a lot of plastics. We made, really what is needed. We what is functionally needed. We attempted to make. It's a very, very unique design. And then we also offered the product in a blister pack, and then very easy to retrieve boxes. So those were the differentiating factors, and then we were able to carve out a market for us. And the last year we sold little more than 2.5 million blades in globally, and mostly in India. And then the equipment division was started when Dr. Namarmaswamy became the chairman of the organization. He wanted us to make affordable green laser. At that time, the lasers were, there were only 2 or 3 companies in India. Most of them were from Germany and the US, and the prices were quite high. And then the number of patients to be treated with the lasers were more. So we started with the green laser. Then we started the digital vision chart. And then we have slit lamps, and then we also have a good phaco machine. And recently, we have started manufacturing portable autorefractometer with the help from a US company. So this is sold for many government programs. So there are many unique products, that we manufacture in the retina. If you take, PFCL, ICG, OcuBlue Plus, and AuroCard, E-Kit, Reti. And in glaucoma, we have room temperature latanoprost, and then RD, which is a Burwell implant. And in cornea, Ozone, we are probably the only manufacturer of Voriconazole. And then we have a K-Pro, which is very similar to Boston K-Pro, and Cornisol. So if you look at all of this product, many of these products, we are the monopoly. There are no other manufacturers. But still, we have priced all these products that our community can afford. And then the EC is one of the equipments. We recently, there's got a lot of value for the community. With the shortage of optometrists and ophthalmologists in the country, instruments like EC, it is portable. It can go to villages. We can refract the people, and then it gives very close to the reading, very close to the subjective refraction. So, if you look at all our attempt, mainly we try to make products which are not available in the market, or which are expensive, or those products which are very community oriented and needed for the society. So this is how we focus, and then try to develop the new products. And then we export constantly to about 160 countries, and then we have a dealer network internationally for in about 60-70 countries. And then we reach other countries either directly to the hospital or through NGOs. So the quality of the eye care, basically the cataract surgery is made affordable. Now we have a cataract kit which we sell for about $10 to $12. It has got everything that is needed for the cataract surgery, from the anxiety tablet till the 1 month post-op drugs, all the intra-op consumables like the gloves, drapes, IOLs, knives, sutures, all of them we put together and then pack, and then we try to sell the kit, which costs about $10 to $12 per case. So this is a very remarkable thing, because in many countries in Africa, they don't have to go to many suppliers. They can just buy one kit from us, and then they can plan their annual surgeries. If they do about 1,000 surgeries, they can buy 1,000 kits from us. So the cost of cataract surgery has drastically come down through our good quality consumables. And then when we entered with a lower price, and then eventually we went for certifications like ISO, which also prompted other manufacturers to follow suit. So because of this, India has become a very, very strong player in the IOL industry. Even China cannot compete with us. Basically, India has got a good number of IOL manufacturers with good quality products. And we also made consistently products that are required by the ophthalmic community, irrespective of the market size or irrespective of the returns on investment. We try to make products which are absolutely needed for the community. So we are a strong organization, about 1,000 people, and then our goal is to reach about 10 million eyes annually with our surgical products in the years to come. And I would like to thank the organizers and all the participants on behalf of our employees. Thank you very much.
Thank you very much, sir. That was a very inspiring journey right from finding the community need to making orphan products to then making devices, and then in some areas being sort of almost the market leader, not just in our country but elsewhere as well. Very, very inspiring. I have two questions for the panel. I'll ask both of them at one shot. We can take them. And then I know Chaitra has collated some audience questions as well. So the questions for the panel is firstly about IOL, the future of IOL technology. We'll get Dr. Ravindra and Dr. Naren to talk about that. What is your wish list? Are you expecting a chip in the IOL? Has it already arrived? And then we'll quickly get Sriram sir to talk on that. And the second question is open for any of the panelists or even the speakers, is that if someone has a product device idea, how does one, and I think this is really the crux of today's seminar, how does one convert that idea into a viable product? Who do they go to? Forget the DBT, forget organizations that can support you. More practical things. Who do they go to? Okay, so first we'll take the IOL question. Dr. Ravindra sir, followed by Dr. Naren, then back to Mr. Sriram, and then the second question.
Good evening. I'm sorry that I had to check in a little late because of technical glitches here. Thanks for inviting me to be a part of this elite, faculty. About the IOL, it's your imagination. So 10 years ago, 5 years ago, we didn't know where we would have landed today. Whether you're looking at optical or rehabilitating a person into the vision, or you would like to have more than what he ever deserved as a human being. So as far as rehabilitation is concerned, it's a lens design that adds on. Multifocal lenses have to go out, have to leave because of the disparity that they have in the actual, what they get and what they are supposed to get. Better designed lenses, which will correct the all the defects of the eye as the God has manufactured them, is probably the way to go. And if you're looking for more innings in that, like are you looking for magnification, are you looking for zooming, are you looking for other things, is the thing of one's imagination. They'll all come inside, but majority of the population would be very happy if they can see distance, if they have good binocular vision, and if they have good near vision. So accommodative IOLs are physically, they're not possible. There's no technology which can bring in the accommodation that the God gave us, but that's one way to look at. Hope I've answered that.
Thank you. Dr. Naren, your wish list for an IOL?
Yeah, I totally agree with Ravindra sir that I also do not I don't believe that the multifocal IOLs will be the future. It's going to be there for a few years and then it's going to die down. I think one very interesting IOL, actually it's from a Swiss company, it's called a Swiss Advanced Vision IOL company, and this company has come out with a concept or a IOL. It's called RTAC, RTASC. So it is, what is done is it's installed an electronic chip inside the lens itself, and what it does is it has solar cells to power itself. So the natural light itself is enough to power it. And the other thing, what it does is whenever there is, let's say, if you're looking at something far away or near, immediately it detects, it autofocuses, and it sends signals to these micro pumps. And this redirects the fluid in such a way that the anterior curvature of the membrane changes, and this focuses. And and it's shown the focus speed is almost 0.2 seconds. So it's extremely fast, almost at the same level of the speed of accommodation of a natural lens. So this seems very, very promising. I think probably it's saying that it's going to be out probably in 2 years. And it seems like a very promising lens, and I feel this could be the future. Yeah. Dr. Anand, your audio is muted.
So sorry. Sriram sir, what is this science fiction, or what does Aurolab have inside the magician's hat? What's coming out next?
What Dr. Shetty mentioned, it is a possibility. It is coming up. This was actually happening for the last 6-7 years. So it was actually started off with a spectacle, accommodating spectacle lenses, and then that kind of knowledge is brought to the IOLs now. because the electronics miniaturization is happening. So because of that miniaturization, now it has become possible to bring an electronic accommodation in an IOL. And many things are also built into that, like, it can also measure the pressure, IOP pressure, and then it can also prompt the user before the pressure reaches a peak, he can put medication. So many things are happening around that on one hand. On another hand, there are companies which is also trying to take such data from these devices and then try to make a business out of it, either for insurance or for drug companies. So a lot of things are happening in the IOL industry, especially with the implantable chip into that.
Thank you. We'll take the next question, beginning with Dr. Devakant. You're a young innovator, and the second question is, if you have an idea, who do you go to? Where would you want to go?
Good evening. So I'm not much of an innovator myself, but yes, I've been interacting with a lot of people who have been innovating. And recently we did a whole, Dr. Anand was also there, we did a whole session revolving around innovators. So this is something that we talked about, whom we should approach. So the basic idea that we condensed was that we wanted to approach somebody who has been innovating, people who have come up with recent innovation. So that would be the go-to person if nothing is happening in my institute. Yeah.
Thank you.
Can I make one comment? Actually, actually, one of the most proud things is companies like Aurolabs and Care Group, all the Indian companies. One thing very unique about them is any innovation, they're very keen. They always try to listen to you, and they're very keen to do something. And the second thing, how different are they from international companies, is decision making. It's simply amazing how fast decision making. If you want some help, quickly the decision making is done. And in fact, if it seems like a really good idea, even the implementation also, they're very ready to immediately do it. So it's really proud to have Indian companies like this.
Can I add, Anand?
Sir.
Well, I had a simple idea, in which I actually reached out to Sriram, and he and I together, along other people, we made that what is available now is called E-Kit. E could stand for anastomosis, could stand for emergency, but that is one kit you need for immediate injection of antibiotics and steroids, including antibacterial, antifungal in anastomosis, or you can use now for your intravitreal other injections.
Wonderful.
So I reached out Aurolab, and they took maybe 6 months to fix it for me, and I will over the market.
Dr. Milen, yes, please. You need to unmute yourself.
Hi. Yeah, I'm responding to your question about where does a person go to if they have an idea.
Yeah.
I mean, I don't have a solution, but I have suggestions. As someone mentioned, that AIOS does not have anything constructive in this area, but we do have sessions where we select best papers from certain subspecialties. So if that could be channelized through the specialty meetings like VRSI and Oculoplasty Society and so forth, and then it goes to a final session in AIOS where the innovation gets an award and it gets respected, as well as it gets its due credit. That is one way you could do it. The other is the institutes like Arvind and LV Prasad, Shankar Nethralaya, Narayan Nethralaya. They could form something like, just like how we have IERG for the research group, there could be Indian Eye Innovation Group where people can come and present their ideas, and you could bring an industry at the same time so that all decisions.
That's a great idea.
I think we have ARC already having an apple tree. I think innovation that there were some really interesting ideas that were presented there. But I think we need to take it to a bigger platform, and a platform that is going to support these innovators. Just coming up with the idea is not adequate. You need to actually keep up supporting them so that they can take it forward. Which brings me to my first audience question, which I think probably Casey can take. Casey, if you can unmute yourself. So this is, I think, Abhijit Khake from Pune. He says, for an individual who has a patented innovative design and wants support to realize it into a product, what is the support and how much equity do the companies ask for? I think this is specific for you, so you'd be good to answer this.
And also maybe Dr. Sivaraman as well, because he's PhD industry entrepreneur. So both of you, go ahead.
Yeah, I think it depends on the overall innovation and what is the potential of the innovation which the company or the investors see. Basically there could be two ways of working, and of course there could be many ways. One is offering an equity vis-a-vis what we do for that particular product, or the simplest option could be actually working on royalty, because the difference between equity and royalty is equity is something which is far-fetched. Unless it converts, it still remains on paper, but royalty is something which you can convert on a sale by sale basis. Now it depends on how fast you can convert and how, because if you're taking equity for a smaller product of a company which has actually got many products, obviously unless that company gets sold out or they go IPO, you can't cash your equity, or unless they decide to pay you. Royalty could be an easier option where you kind of give your patent license and then take on a per sale basis. I would say that is the most practical solution today. But if you really want to go the equity route, then it directly depends on how much the innovation is valued, what is the competition, and what is the overall potential of the whole thing, and what the investors see, and then that is converted into equity.
Dr. Anand?
Hi, I think I'll just add one other point to what Casey talked about, which is sometimes the readiness of the innovation is something that has to be mapped to the right type of funding. For example, it may be too early to think in terms of a transfer to a company through equity, etc., or royalties. There could be, for example, a stage of development where you could use funding from grant bodies, like we took Wellcome Trust funding when we initially got started off. Bring it to a point where there is commercial relevance as a result of actually showing that there is a business around it. At which point I think the discussions with the companies would make a lot more sense, because it's been sufficiently demonstrated. That's more from the perspective of most innovators then being able to have those conversations at the right time.
Right. I think there is one more question where Dr. Sriram can probably take. This is, are there, Dr. J.K. Reddy wants to know, are there any ophthalmologists in the world who with their invention or innovation have made good profits, like Slade with crystallines? I think since you've come up with so many innovative and they have been, profitable and important for the community itself, would you like to take this question, sir?
There are some ophthalmologists who have invented and then they have sold their invention to companies and then made money. But that is not so much seen in India. But we as an organization, we have worked with many ophthalmologists in bringing out the products like Dr. Das mentioned. E-Kit we developed with the help from Dr. Das. Last year, we launched a product called XNet that we worked with one of our doctors called Dr. Prabhu Bhaskaran, who worked in Aravind Pondicherry, now in Chennai. So we worked with him. And then we also, last year, we brought one another product called amniotic membrane inserter called AMSET, working with Dr. Bhaskar Rai Chowdhury from Calcutta. So we have, like Casey mentioned, we have a royalty route with which we work. For all the development, we invest the money. And then, if it succeeds, then doctor gets a share. So that's a route. So we are open to work with any ophthalmologist in the country if they have a good idea. And the patent, we don't encourage unless there is a big market and it has to be protected, because patent itself, there is a cost to it. And patent is a territorial, you have to cover all the countries, which is very, very expensive, unless you have the ability to market and if, whether the market can return that kind of investment. Otherwise, like amniotic membrane inserter, we discouraged, going for patent because it's a very, very small market, not worth going for. So all the innovations need not be patented, but definitely can be brought to the market, and then we can see some financial interest in that.
I think that's an extremely nice thing that you said, that anybody who has an idea, you can actually help them implement it. And I think that's an open invitation to all you young innovators and entrepreneurs. I think you can carry on, Anand. I'm done with questions.
Thank you. So we've finished our keynote veterans. We'll now move to the best actor section. Again, from that same spot there is going to be Dr. Kim. Dr. Kim, thank you, Dr. Sriram, for that very enlightening talk and for fielding all those questions and giving us all the encouragement. Dr. Kim is the Director of IT and the Chief Medical Officer of the Arvind Eye Care Systems. Everyone knows him, especially in the retina circles. And he's really done a lot of innovation in artificial intelligence and has worked with global giants and has taken Indian ophthalmology and AI to a global level. So we've sort of titled his talk as Google Diaries. And Dr. Kim, please take over.
Thank you, Anand, for inviting me for this important webinar and giving me this opportunity. I think I've been talking too long about AI for quite some time now, but because of my passion for this particular, especially it started with this diabetic retinopathy challenge that we've been working on for the last 2 decades, with Dr. Nam pushing it, driving the whole thing, and somewhere along the line, we have been doing this diabetic retinopathy screening for quite some time, because as you know, in our country, this is really way in the back where not many people actually do this screening for diabetic retinopathy, and it really is a very clear use case for using it in telescreening. So that's how we started, because we have clear guidelines on how to use these images for diabetic screening. And so in 2002, we started with, of course, ISRO's help. We and Shankar Nethralaya also at the same time were developing this kind of a screening program wherein our team would go use a satellite connectivity to get the retinal images of patients, diabetic patients, who have been pooled there and then sent to a reading center with the report being sent back to these mobile vans. And later, these mobile vans went away, and then we have been using these fixed facilities. That's a diabetology clinic, where we call what we call as an opportunistic screening, wherein the diabetic patients walk in to these diabetic clinics, and you have their technicians take a picture and send it to us. We've been doing this for quite some time with a turnaround period of 1 hour when the patient gets his report back. But what are the challenges is to have trained graders to make a retina specialist to read these images is a waste of their time. And of course, the time delay is sometimes 1 hour to 2 hours, or in many places, it's actually over a few days that they would give a report back. And most importantly, the inconsistency in grading when this image where we had subjected the same retinal images to 30 different retina specialists, the grading that happened was so varied and so inconsistent. It really shows the inconsistency in the grading, the manual grading, by whether it's a retina specialist or by a trained grader. So we've been working with this diabetic retinopathy, and way back in 2004, I was hearing about this image processing, and that's when I got excited, thinking that we could use this, and so started working with various universities and especially many engineering institutions, and it would always end up with getting a paper published, and beyond that nothing would happen. So when Google came out in 2013 and asked us, at the same time even Shankar Nethralaya was approached, I was really excited, because there now you can actually have a product, because they are not going to be interested in publishing, but more interested in getting a product out, being a commercial company. So that's how the ARDA, the Automated Retinal Disease Assessment tool, was born. And we've been able to use it for grading the diabetic retinopathy, and also for DME grading is the current status with which this is being used. As I said earlier, we would take a fundus picture, send it over the internet to a reading center. and the reading center would generate a report, and then this report is sent back to the clinic where the patient can be counseled about the eye status. So with the AI tool, this was replaced with the turnaround time of 1 minute, no need for sending it. Now we have offline, online AI tools from various companies. Of course, Google says what we are using currently is an online tool which we need to upload the image into the cloud, and then we get a report within a certain period of time. So this has made a lot of things much easier and is not dependent. So this article actually came out in 2016, the first article on the on this algorithm, and just for your information, that this was chosen as one of the top 10 articles of the decade by JAMA, and this was the only article in ophthalmology. And not just, there were a lot of other new discoveries which came out of these studies when they were looking at the various images, where we were able to predict the cardiovascular risk, 5-year cardiovascular risk factor by just looking at the fundus images. Imagine predicting the gender of the patient, age of the patient, or the HbA1c levels of the patient just by looking at these images. Now, of course, all these need validation, needs a lot of data. And this is happening as we speak. A lot of work is going on how this can be just a retinal image can be a predictive tool for so many other systemic conditions, not only for diabetic retinopathy. So subsequently, we did again at Shankar Nethralaya and Arvind, we did a validation study of our own using this AI tool with our regular, how it performs in the clinics, and the sensitivity and the specificity was very high, as you could see, or more than 90% in both the institutions. I don't want to go into the data, but after that, since last 6 months, we have implemented it in real time. So after the CE certification was used, currently we use it in 3 use cases at the diabetic clinics where we were outside of Arvind, where we are screening the diabetic patients for retinopathy. We are also using it in our vision center, some of our vision centers, where this is the AI tool actually predicts or identifies, diagnoses the diabetic retinopathy. And we are also using it in our Madurai clinic as an assisted read in the general ophthalmology clinic. So today we have done about 15,000 plus cases are being read. And of course, we are doing some validation in the background for these studies, though the data that comes out of it, or whatever the report that is generated, is what by ARDA, or the tool is what is used for treating the patient or guiding the patient, not treating, but guiding the patient. So this is just one effort, but there are, as all of us are aware, there are so many attempts being made using AI. Diabetic retinopathy was a low-hanging fruit, so everybody used, or many of the companies are still working on developing this AI tool, and many of them have developed, and there are quite a few tools which are commercially available. Anand is using an offline tool in his camera, and so quite a few are there in the market today. But it doesn't stop there. There is a lot of use cases using AI where it will make a huge change. The OCT, the DeepMind I developed how to read the OCT scans using AI in UK, and this was bought over again by Google, and they're working on that over a period of time. But they're also looking at how you can use, even doing a prediction, just looking at the retinal images. Can we predict early on that this retinal image, the patient having an early diabetic retinopathy, could go in for a proliferative disease just by the pattern in which it presents? This is something, some interesting data is coming out in these studies, and we may be able to predict the future of those patients by just looking at the fundus images. This is making it more interesting. And this is not just that, but just by mining our medical records, we could do a lot of predictability, outcomes predictability could become a day-to-day factor. And I don't know how many of you are aware that online consultations is being done in the US. The NHS is in the UK, by the NHS is trying in certain areas, and this is widely used in Ethiopia by a company called Babylon, where they are able to, the patient just have to call for any problem. They just call the NHS, and then the system basically asks a few questions, and based on the questions, it gives them an answer asking them to use this certain medication or use this certain things and then come back next time. The next day, if the patient doesn't call back, the system itself calls them, asks them about their progress, and if they are not improved, it directly connects them to the concerned specialty based on the symptoms. And this is all done in the background using just AI. And this may become the way in future where access to care is going to be very, very difficult, or it's not there. So as I said, this could become a very good clinical decision support tools. And by mining the medical records, we can improve the diagnosis and predict outcomes for treatment. And I see it today as a very important triaging tool in many of the primary care centers by just using AI tools in the near future. So that's my idea or interest at this point of time, how we can use these AI tools as in these primary care centers to triage patients, so that patients who have serious problems actually reach to the specialist and do not overcrowd the retina specialist or the ophthalmologist offices with simple problems. So thank you, Anand, again for letting me share this work here.
Thank you, sir. Thank you very much. And fascinating world of AI. Imagine looking at an image and then the image itself telling you what gender that image came from. Amazing. So we'll take a quick take on AI, and is AI really a threat to doctors? Let's get a word in from Dr. Tirumalesh. He's been working on AI, and then a very quick word from Dr. Anand Sivaraman. He's been doing some AI work as well. Is it a threat to doctors because AI is doing what it's doing now?
I don't think it's a threat. Probably it would help in far outreach of medical services, and probably reduce unnecessary crowding of the retina clinic, like Dr. Kim said. And the other part that we should look at it is, it's great that we're all excited that AI is going to give us a lot of input into the disease and also input into their systemic problems, but a lot of data validation is also required. Say, for example, the AI that we are developing indigenously since past 3-4 years, we have not been able to replicate or identify whether the patient is male or female. Second part is we are not able to tell whether a particular patient would be of higher cardiac risk beyond the measure of the arteriosclerosis grading that we already know clinically. However, when it is linked to what is called as data mining, then it kind of gives you more information. So what current generations AIs are doing are, they are looking at cross-sectional data along with medical record data mining to predict an outcome. What would actually tell whether this is going to be true or not is if we collect what is called as longitudinal data and see whether we can match that and validate that. So that is going to give a more truer picture when it comes to applications of AI.
So doctors still have a job. Dr. Sivaraman, a quick take on that.
I think there are two things. One is with respect to, is it there to replace an ophthalmologist? I don't think the intent would be to look at it that way. Today, when one thinks in terms of public health screening, even for diabetic retinopathy, in a general population for opportunistic screening, you're possibly going to have more than 70% of images that are going to be negative. The way to think about it is, would we want to use an ophthalmologist's time for them to say that that particular person is negative, or can we actually have automation help them there? Especially when there are significant low number of ophthalmologists as specialists available per patient. That's one. Second, I think all of us in this space also realize that we are also very cautious in the way we deploy this, not only from a regulatory perspective, but it's also one that requires a significant amount of monitoring. For example, just as Google's come to India to look at how their algorithms that have been trained on a certain set of images work in India, similarly, we've been looking at what are differences as a result of ethnicity, as a result of using images that are trained, let's say in India, and an AI that's been developed with that, but how does it perform in Europe? How does that perform in the US? I think there are interesting research questions that also need to get addressed. I think long term, it is only going to make it much easier for ophthalmologists to be able to focus on treatment, focus on patients most in need for treatment. And I think that's how we would initially look at it as triaging before we think in terms of anything more than that.
Thank you. Chaitra, you want to take the audience questions forward?
Yeah, there is one question for Dr. Kim from KL Nisha. She wants to know what would be the compromise between sensitivity and specificity in such AI-based tools.
What is the compromise? I don't think we are, because we have a high sensitivity and specificity in Aman, especially when compared to the manual grading that has been happening. So if you look at it, I don't think we are compromising anything in this. But we also have to understand this is not a diagnostics tool. This is only a screening tool that is being used. So we have to be very clear about the difference between a diagnostic tool and a screening tool.
I think that answers that question, and we can thank Dr. Kim. Really inspirational work this is, and you've put us on the map with the work that you have done. Thank you so much. We'll go over to the next speaker. Great privilege to introduce Dr. Suven. He's the director of Nayan Eye Center, Kolkata. He's also the director of MedInvent Devices Private Limited and the inventor of the BHEX pupil expander, which all of us know about, and it's hugely popular. Over to you, Dr. Suven.
Yeah, thank you, Chaitra. I'll just try to share my screen first. Yeah, is my screen visible on your screens?
Yes, please, it is. Need to go to presentation.
Are we good?
Yes, perfect.
Okay. Thank you, Chaitra, for the very kind introduction, and Anand and the entire team, the backend team, for all the support. Yes, I'm Suven Bhattacharjee, inventor of the patented BHEX pupil expander, and I'm also a director and shareholder of MedInvent Devices Private Limited. Though I'm here principally to inspire young innovators, I believe by the time as I speak to you about pivoting and improvisation, each one of the viewers is going to discover the innovator and entrepreneur in themselves. So just a little bit about the BHEX. We have issued patents or granted patents in 17 countries, including US and lots of countries in Europe, and of course in India. And in India, we launched in 2017, and it costs less than 20% of the cost of the imported Malaysian ring. It's much easier and safer, as many of you use it and tried it firsthand. And we believe we are leading the pupil expander market in India. We are exporting to about a dozen countries, not a great big deal, but yes, we are doing some work over there. So as in basketball, pivoting is basically we keep one foot stationary or anchored, and we try to look for exits, and when we find the opportunity, we just score the basket. So that's all. So startups and businesses use this term often, and a pivot is a change in strategy without a change in vision. The vision remains constant. But I'm changing the context, and I'm applying it to the innovation cycle, because I believe that innovators are pivoting at all times, at the design stage, at the prototype stage, at the validation stage. Innovators are always pivoting. So the typical roadblocks would be the prototyping and patenting, the clinical testing or validation, and the commercialization. So let's take a look at the BHEX as an example. So the vision was a simpler, safer, and economical pupil expander to be used by all. The BHEX design had to be simple. So we had an open ring, single plane, clover leaf design, looked very nice on paper. Well, easier, why the open ring? Because it's easier for insertion and removal through a side port. Well, good enough. So the next step was obvious. So phone a friend. Well, the typical question I was asked was, have you filed a patent? And when I would answer in the affirmative, their answer would be, sorry, that's too much of hassle. So I didn't find a partner to work with. I soon realized that when we hit a roadblock, it's always a good idea to kind of step back, and we will see the detour. So for me, it was like, do it yourself. So this is the first prototype, the drawing, plastic sheet cut out. Looks very elegant over here on the slide, but when I saw it under the microscope next to the eye, it looked obnoxious. So we rejected it. So the art of pivoting is basically turning the unforeseen challenge into opportunity. So I needed something springy material, the right amount of resilience and flexibility. So nylon kind of ticked all the boxes. So a nylon suture would work very well. But then how do I get to bend that nylon and stay in that shape to serve my purpose? So I went and met a plastic expert, Mr. Maheshwari, a very kind man. So he asked me a question. Does your wife bake cakes at home in an oven? I said, very much. I said, why don't you go home, wind a piece of nylon suture on a nail, on an iron nail, and toss it into the oven, and you'll have your spring. I said, well, that's good. So my wife's kitchen oven was the birthplace of the first butter charging or the BX. And this is the first spring I made out of nylon suture, very sentimental about it. So what we did was we converted this question of how into a turning point, and then there was no looking back after that. So this was prototype 2, 3/0 nylon, but then we cut it into sharp ends that looked very dangerous. So well, we rejected that too. And then we went on to kind of making eyelets into those legs. And we thought, well, that would be a lot safer. But then we did use it. It worked, but it was gross. 3/0 nylon, it was an open square. It looked huge inside the eye. So then came the unanticipated problem. The legs came close together because of the constrictive forces of the pupil. So we were not factored for that. So then now we went on to more notches and flanges, and we also blunted the tip. Well, that worked well. Legs came closer again, and sometimes even interlocked. That was very embarrassing during surgery. So pivoting is both an art and science. Adjust and leverage unforeseen circumstances to provide positive results. So then came design 7. Of course, we went to about 17 or 18. The house-shaped hexagon to counter the constrictive force of the pupil margin. This worked pretty well, but still the success was limited. So now we realized that we needed to close the ring. But unfortunately, the literature says that it's very difficult to join nylon end to end. It's about 5/0 nylon, that's pretty thin, 0.1 millimeter. So it requires ultrasonic welding. I didn't have the means. So I said, okay, let's try. The $5 Fevikwik worked very well, proved all literature wrong. So we got a closed hexagon with glued ends, 6 flanges, 6 notches, started using medical device grade cyanoacrylate, and we had our trials in place. There is sunshine after rain, always. So we had our own clinical trials with the 4/0 nylon, and then we moved on to 5/0 nylon, which we sent to global key opinion leaders. But then this was still handmade. I made about 300 of these. For 1 hour, it just take to make each one of these, and there was a 30% rejection rate. It was real hard labor. So now we wanted to go in for commercial production. We didn't have too many friends in the industry who would help us do this. Well, it was do it yourself again. So lots of research for months together on 3D printing, steel wires, beryllium copper, stainless steel. Nothing really served our purpose so well till we hit the final material which we required, and that was polyimide. It's one of the toughest plastics on earth, and we had a commercial version ready, and we started shipping them. And we started using them with a manipulator. Initially, there were holes which could be manipulated, but then the manipulator was a problem. It did not hold the flanges securely. So despite filing each of the thousands of manipulators that we sold myself, I myself ripped the anterior capsule in one of these cases. So we abandoned that, and we recalled all the manipulators. So we needed the 23 gauge forceps to do this job. The price was a big issue, and there was a huge cartelization in that. So we took a very bold stance. We approached one of the distributors and told them that if you do not bring down the prices, we will start selling this at half the price, and we will take over the business from you. The threat worked, and we brought down the prices all India to 50%. And that works for all VR surgery, forceps, and everything. So pivoting is avoiding considering the unwelcome situation of failure, simply because standard methods or tools didn't work. Now we had to come to the housing. So the BX is very thin, very flexible, and it can jump out of the housing. So we need to deliver the device. We needed the surgeon to have it delivered at the incision. So, and we needed a transparent housing. So we got a polyester sheet cut out in shape, and the flaps were folded. It required a lot of manual labor. Well, that was all right, but then there was a problem. It failed the drop test outside the packaging. Once inside the packaging, it didn't matter. We could ship it and it would never come out of the ring. But then if outside we dropped it on the floor or something, it would, from a meter's height, it would just pop out of the housing. That was not acceptable. So we moved to the current housing, which is a molded housing with a transparent lid. And this is a very robust housing. It passes the drop test every time. So then we came to the pricing, and well, initially I was naive, so I depended on the distributor. I'm not a business, we don't have a business background. We need training for that. So very soon we realized that no, we have to take charge. So we made in the devices to control, and we brought down the prices to about 50% of what we were selling initially. And we recruited more distributors. We started exports. We expanded our markets. So to pivot is options and solutions should be the focus, not pre-existing models and processes. Now, in all this process, did anyone notice something? An eye surgeon turned into an innovator who then turned into an entrepreneur. And trust me, the journey continues. And I'm not even halfway through. So there is no perfect way to innovate. There is only a passionate way, and I always believe in that. Thank you very much for listening.
Thank you, Dr. Suven, for sharing that very inspiring story. And I think you said it all in your last slide. There's only a passionate way. A quick question, actually, this one is more for you, but the panel is welcome to chip in. We understand the importance of filing a patent early. But if there is let's say there's a change in the design, and you've thought of something down the road, how do you provision for that once you've already filed for an expensive patent?
Yes, see, the patent rules allow you certain modifications. So as long as you do not deviate from the initial design, the core design. So there are two ways of doing that. You could file a provisional patent initially where you have the diagrams and the description. You do not file the claims. And you could go on, you have now 12 months to file your complete specifications, which would have the claims. So there you can add your diagrams, you can change your description to a large extent, not deviating from the main core design that you offered. So we can't deviate too much over development. Yes, but you can add as we go on.
Great. We have a question from one of our speakers itself. Dr. Divyansh is asking, all medical devices in India have to be regulated as drugs. Medical Devices Amendment Rules 2020. So can we try a surgery or surgical instrument innovation before clearance?
Mr. Sriram probably is more competent to talk about that. But then, yes, there are certain devices called notified devices, which was there till the Drug Regulation Act of 2017 came into place. It was supposed to start off again this year, January. I think this will get deferred by another 1.5 years. There's a time period for coming in. So as of now, devices are in kind of the cusp area where they are not fully regulated, but they will be regulated. But for trials, you can always have institutional review boards clearance, and you can have ethical committee clearances and use it, especially if it's a similar device. If there's a predicate device, you can use it. If it's something de novo, it will require a lot of clearances.
Thank you, Dr. Suven. Before we sign you off, just the single most point that you would advise innovators and entrepreneurs, just one.
And the two. So that's one is never lose sight of your vision. And two, there is always sunshine after rain. There is always sunshine after rain.
Thank you so much. Thank you.
Thank you.
Let's move on to our next speaker now. A friend, we call him Casey, Mr. Chandrasekhar, who was the founder of Forus Health and is currently their CEO. And he's going to talk about all the devices that he's taken onto stage when there were none in that field, and working in some niche devices. So, Casey, over to you.
Thank you, Dr. Anand and Chaitra, for giving me this opportunity. And it's a great opportunity to be part of this ecosystem and present. Can you see my slides? Yeah. You can, right? So I have repeated this many times, but again, this has become a habit. So we had no knowledge about ophthalmology medical devices, and we came into this business because we were inspired by Arvind Eye Care. And we worked on a similar mission of eradicating preventable blindness or avoidable blindness. But basically, when we started, we knew that there were several problems, which were the order of magnitude was very big: cataracts and diabetic retinopathy, refractive errors, ROP, ARMD. And we thought, given that we have less number of ophthalmologists, the way to approach this is do a preventive mass screening for problem identification and push them into the treatment ecosystem so that they don't reach the hospitals too late, so that it is difficult to kind of save their vision. So when we did an analysis at that time, basically we found that there were devices which were really good, but they were expensive and bulky, and only specialists had most of them. And it also required a lot of specialist technicians to operate this. So we zeroed in on making devices. So want to just reiterate that, we were not devices people. So it was not, okay, it was a very de facto option to make devices. Because of the problem and the understanding of the problem, and we found that there was a gap, we felt that making devices may be appropriate and add value to organizations which are working in this preventive eye care space. So the only thing we kept as a specification was that it should work on imaging, and it should work on undilated pupil. It should be obviously affordable and portable, and more importantly, easy to use, so that we kind of de-skill who takes pictures. And finally, it should be accessible to in primary care. So that's how we started. And the first thing we came up was with a non-mydriatic digital fundus camera, the Trinetra series of products which we made. So the first one was the Trinetra Classic, and then we made the next version called the Trinetra HD, which is already, you see in picture, is already COVID, being used in COVID conditions. And then we are launching another one in a couple of months called the Trinetra Ultima, which is basically a higher resolution, autofocus, and which will also meet some of the international customers' requirements. All along, we basically focused on, making it compatible, making it, usable in communities, at the same time, ready for telemedicine. And today, the product made in India is there in 36 countries, and we have crossed 2,000 plus installations of only these series of products. So I obviously, with very minimum knowledge, could make this primarily because we understood the problem, and the guidance given by Dr. Kim and Dr. Nam and people from Arvind Eye Care. And also I fondly remember the motivation Dr. Taraprasad Das gave us when we were nobody. So we could make that and, kind of proliferate that into the market. The next thing which we focused on was wide field imaging for neonatal screening. And again, there we found that there was a need, and the equipment from Clarity, which was at that time almost like $120,000. We felt that if that can be made at a 1/5 of the price, then probably we will be able to, screen more number of babies. And what was also happening on the background was that, the number of babies surviving which were preterm in India started to improve because the neonatal care started to improve, and there was a need to screen these babies. So with the support of Dr. Anand and Naren Netralaya, without which we would have not even understood what is ROP, we were able to roll this product, Trinetra Neo, what we call. And today this product is more than in 120 installations across 15 odd countries, and very proud that NHS has been adopting this device in UK across 15 odd installations, and they have not been using Clarity. We also are making the next generation product, which will probably be number one in terms of the specifications globally, which will have 130-degree field of view, a 20 megapixel camera with fluorescent angiography integrated, which we will launch in a couple of months from now, or maybe early August, September. And this is already started on the validation stage. the good part is this product actually went through the complete 510K application and got cleared, and also got the CE. This product was actually awarded the BIRAC grant and also the certification for Forest Health, Narayan Nethralaya, and IIT Madras. And this product also won the Marico Innovation Award way back in 2018 for the best innovation. And that followed up by a book which actually talked about 7 sutras of innovation, which actually has great organizations like ISRO, Tonbro, and all those companies. And they also included Forest Health as one of the 7 innovations. So it's a very proud part for all of us. And we can make a product from India and make it #1 in the globe. So today, the Trinetra Neo is probably world #2 in terms of the number of installations. And probably in the next a year or two, we'll probably become the #1 in terms of the installations we do year after year. Again, not to forget, phenomenal support given by Dr. Anand and the team from Narayan Nethralaya for making this happen. And that really helped us to take this product globally. The third thing which we focused on was refraction. So one thing we were very clear was that we focused on only 3 areas, large areas: retinal imaging, neonatal imaging, and refraction. These were larger problems. And we wanted to just focus on that and continue to go into depth rather than, kind of doing many things. We launched our wavefront aberrometry-based refraction called the Avero, and already crossed about 100 installations of this. It will be the easy and fast autoref which you can take to homes and start screening. And we are also launching our next innovation on probably one of the we have kind of applied to more than 3 patents on a wearable phoropter, which you can actually do subjective refraction using this wearable phoropter and control the entire thing using a Android phone. So without changing lenses, you can do accurate subjective refraction. So this is likely to be launched in a couple of months. So these are products what we made. But I think one thing which I want to reiterate and continue to reiterate, I think we created products to solve a problem. And I think if you really ask us, have we reached our mission? The answer is no, because maybe we have touched 6-7 million lives, but that is very restricted because we are still making devices. So only when you broaden the facet and start going deeper, how do we proliferate into primary care? How do we proliferate into diabetic clinics? How do we proliferate into optical shops? You have to do innovations which is in deployment. And I think most of the innovators don't want to do this because it is not glamorous. Saying I have a patent, I have a product, I have a thing, that's very glamorous. But for us, what is really glamorous is if we can actually multiply the effect of the number of people we touch. And in order to do that, we strongly believe that we need to take these products to the last mile. And in order to take this to the last mile, we need several components which are very important, which is building this digital health platform. So we worked on this, and over the last 2 years, we have been able to get something going, and we already have like 2 million patients using this platform. And what it does is it connects to devices, and which is a very significant difference is the moment you take this kind of an approach, you not only just connect your devices, but you also connect other devices. Because at the end of the day, while you're passionate about your innovation, your product, the cost what you're working on is much more bigger than your product. So when there is already an existing product available, you need to kind of connect to that. You need to kind of create some new products which may be required for home screening or which is required for a different kind of a purpose. And that is what we are now trying to do. So we kind of integrated this whole thing. So it will do teleconsultation. It will also upgrade itself into a disease management profile. It will have built-in EMR, which we took from a third party. And the other important strategy which we adopt for on the AI side was that we will develop something complementary, but at the same time we'll work with all the AI modules which are available. So the best part is, the digital platform connects to multiple AIs and it can actually do many things. So because the last mile is the key, and that is where we felt we were more connected, we were more passionate about. And I think that innovation excites us much more than our product innovation. Not that it doesn't make us happy, but I think because you make a product and sell, maybe you're touching X lives. But when you're creating it in a platform and delivering it, we are able to touch 10X lives. So I think if we can work more on this and reach people, and with the support of ophthalmologists, we can go and touch lives which is outside the purview. And this is obviously going to be triggered by the post-COVID era, because consultations outside hospitals are going to become a norm. And I think if we can kind of work with this and kind of develop and deploy this, I think that would give us much more satisfaction. So that's all I have, and thank you very much once again for the opportunity. And any questions are there, I'll be happy to take.
Thank you so much, Casey, and congratulations on the journey of Forus. In less than a decade, you've put the company and the country on the global map. And personally, of course, we've been part of your journey, and it's been wonderful. And we're so happy that you are looking at that final mile of taking this, not just the innovation, but the entire, not just the product line, but making it available and accessible to where it's needed most. The question is there, but I think it's open to everyone. We'll begin with you quickly, but anyone who's dabbling with telemedicine is welcome from the panel to take the question. You briefly, Casey, mentioned about the teleconsult link that you are creating in your devices. Do you think that this recent scramble for doing telemedicine among all hospitals in the lockdown period is there to stay? Do you see it as a trend? And do you see patient behavior changing now and even after lockdown? What's your gut sense of this thing?
No, I think one thing is, I think there is a scramble to do, and I think everybody wants to get into it. They will really understand how effective it is only when they start using it, because, saying that I am doing teleconsulting is something which everybody wants to do. But then the reality is that how effective it is going to be, and you need to do many things in order to make it very effective, because you have two sets of patients who are coming for follow-up and new patients, right? Patients for follow-up, if you have EMR and other things, you will be able to kind of consult them far more better. But if you're getting new patients, I think the doctor will find it difficult to consult unless it is for a very simpler problem where he can kind of, otherwise without imaging data or refraction data, it is very difficult for the doctor to consult, despite the fact that he will see the patient's place. So I think that is what my take. I think it will definitely have an impact for the next 3-4 months, but it will kind of go down after some time. But I think what it will leave is, today, if it is like 70% teleconsulting is happening, it will kind of settle down at about 40% going forward. And I think it is up to us to use this behavioral change far more effectively, because a lot of times I've been asked this question: what is your biggest challenge as an entrepreneur? I think the biggest challenge as an entrepreneur is actually the behavioral change in people, right? That is completely not in your control and is very expensive to do. So I think COVID is kind of going to drive that, and hopefully we can use that to the advantage and kind of deliver care.
Thank you, Casey. That's encouraging. Chaitra, do you want to take it forward?
Yeah, Casey, there is one question for you. What is the hardware innovation that you have that can support the use of AI? Kaushik asks from Bangalore.
I think, basically how we designed our systems was that the hardware which we make was fundamentally optics and electronics. And we kept the entire, the overall performance, the movement and the collection of data, everything on a laptop or probably a dedicated computing. So obviously all the AI performance happens. And the reason for doing that is primarily because, year after year, these, processes are going to become better and better. And, if you have a dedicated computing, then you're actually restricting the use case, right? Now, when you have it on a laptop, you can do anything. It runs on the laptop. Sometimes it runs on the cloud. So you are able to kind of perform all of that outside. So, as AI keeps going more complex, we can continue to work on it because we are doing outside the fixed device.
I think, Anand, you can also take that question, because you're already working on a lot of AI at Remedio, and we have also partnered with you on that. So would you want to take give some inputs on that?
I think the key point to add to what Casey did is, There is the regulatory part to it, and then there is the clinical part to it. The regulatory part is today, FDA is mandated, let's say, certain minimum sensitivity and specificity if one looks at it globally. And their approvals for AI is linked usually to the device on which the clinical trials were done. So I think there is, therefore, the only other company that I do know of called INAC is actually attempting multiple devices simultaneously, but it has not been easy as well. So I think what's going to happen is it may take a little while until global regulatory agencies may consider AI being used with multiple devices until significant confidence of clinical trials comes in. Given that I think it's early days in terms of research on variation in devices and the impact that the image quality and image features may have. So I think that's where things are right now from a clinical perspective. And I think that's where, to Casey's point, in a place like India, if let us say the way we define the problem is to say that, look, the sensitivity is what's most important rather than the specificity, it then results in maybe many new ideas. And this is where I feel we need not necessarily follow what the US FDA or MDI Europe is. I think from clinicians, if we are able to, if the clinicians in India are able to define what may be keeping the best interest of the patients and the public health in mind, then we may be able to come up with other modalities. But today, there is the restriction on the basis of the way the regulator looks at it today.
Right. Dr. M.S. Ravindra also has a question here on the chat. He's asking, how about AI helping clinicians in their day-to-day work, like differential diagnosis and treatment options, etc.? So Anand, since you're also into a lot of clinical work, you can probably take that question.
I can talk about maybe some examples of what we've seen outside of the work that we've been doing at Remedio. For example, the entire aspect of using AI in clinical record taking. SNEC, for example, has been working with IBM Watson and looking at the use of AI in being able to fill up and record clinical case history so that it increases the time that the doctor, the specialist may spend with the patient. So there's been, with the kind of advances in NLP as well as text-to-speech and speech-to-text technologies, a lot of that has been happening as well on the side of EMR and AI. There's also work on the use of AI, though it's still in research. It's not something that's been commercialized as yet, in looking at longitudinal data, as Dr. Thirumalesh talked about, and seeing whether or not there is impact of treatment or impact as a result of different types of treatment. But most of those are currently in early research, not necessarily those that have passed through regulatory to come to commercial use. But there's a lot of exciting work that is happening there. There's an interesting area of transfer learning where, for example, while diabetic retinopathy is very well defined, glaucoma is not a very easy problem. So there is a lot of work that people are doing in assisting ophthalmologists and looking at OCTs and fundus and looking at maybe changes that may be marked out to look at maybe multiple units of data. So most of these, however, are in research at this time.
Thanks, Casey and Anand, for that input. I think we'll now go to our next speaker, and that is Javed Ali. He has already put his affiliations on the slide. So I will talk about a few other things about Javed. He's one of the rare recipients of the Experienced Researcher, Senior Alexander von Humboldt Fellowship Award. It's one of the pinnacle awards in the research world. Other than that, he's also recently been awarded the Shanti Swarup Bhatnagar Award in Medical Sciences, the highest multidisciplinary science award in India. And he has close to 400 publications at this time and has delivered lectures in 300 conferences and 12 keynote addresses. He popularly conducts instruction courses and live surgical workshops and over 30 national and international awards. Javed, honored to have you amongst us, and over to you.
Thank you, Chaitra. Am I clearly audible?
Yes, you are.
Yeah. So thank you, Chaitra and Anand, for giving me this opportunity to be speaking in this August group. I would be speaking on the top 3 innovations that we did in dacryology To begin with, I bring to you the greetings from the Institute of Bacteriology at the various campuses of L.V. Prasad Eye Institute. These are my financial disclosures. So before I go, as Dr. Das was asking me in the morning, I think what summarizes innovation spirit and the concept of innovation, I can summarize in what Faraz had said long back. And I quote him, he said, "हम नींद के ज़्यादा शौकीन तो नहीं फराज, हम नींद के ज़्यादा शौकीन तो नहीं फराज, पर कुछ ख्वाब ना देखें तो गुजारा नहीं होता." So this very much summarizes how innovators are predominantly working. So the overview would be only 3 topics that I would be touching today. Number 1 would be navigation and stereotactic enabling of the endoscopes. Second would be the use of coronary angioplasty balloons for BDCP. And third, I would take you through an innovative technology that we use to redefine skeletal muscles anatomy. This is, you can see Milind in the picture. This is just to give you a brief of what stereotactic surgeries are and what is navigation guidance. This is the navigation guidance machine. What it predominantly does is it acts like a GPS. So if you use its tracker, it will intraoperatively in critical location, it will give you radiological information about the location, precise location where you are at that point of time. So this is one of the trackers that you can see, which is used to track the patient. Then we enter a lot of radiological data, CTs, MRIs, CT angio. There are a number of things that you can use it. And then the computer asks you to, when the patient is on the table, the computer asks you to touch certain points with which it then aligns itself to that radiological data. And then it gives you, when you're working in critical locations, like for example, this picture, if you see, this is, we have opened up inferiorly and we are touching one part of the lateral orbital rim which is fractured, and it gives you exactly where you are located. And you can have a lot of windows, axial, sagittal, coronal sections of CTs, MRIs, or a 3-dimensional like you see here. So this is, broadly how a navigation guidance acts. So we had introduced a term called as image-guided bacteriolocalization that encompasses all these stereotactic maneuvers that can be utilized for lacrimal sciences. Now, this is a typical navigation-enabled lacrimal OR that you see. You see the endoscopes here, which we commonly use for lacrimal surgeries, and the second is the navigation machine. So now, what was the need for us to innovate about something? Now, all over the world, wherever these navigation machines are used, these are the different kinds of trackers that are available. Now, for us, what was happening was that, well, we can go ahead and use it along with the endoscopes. Like this is the endoscopes that I'm using, but we could also then need to intermittently stop at every step, take a tracker into the nasal cavity or whichever area that you're operating, and then find it, and then again come back. And that usually, the expenditure of these surgeries, other than the use of navigation, was also enhanced because of the amount of time that it was taking. So then we thought, why not enable our endoscopes to use navigation? And most of the trackers that we were using could not be combined with a endoscope. Then we found something which is called as neuronavigation stillet that the neurosurgeons use during the shunt surgeries for navigation. We thought, well, this is not exact, but we should be able to somehow fit it over the endoscope in a way that the endoscopes then becomes navigable. And this is what we did. We did very simple design of that one, where we used the tracker just behind the edge of the camera, and then we used the neuronavigation stillet right across till the tip. And what we did while doing this is that we did not want it to come beyond the telescope because that could harm the tissues. So we kept it in line with that, and we used a simple tape here, if you can see. And this tape, we made sure that it does not touch any of these red areas, which has electromagnetic coils, which are going to help you with navigation during the surgery. And then this is an example of how we were using it. So what we did was, this was how we used to do it before. This is the tracker, which you can see is quite thick. Now what is happening is this hand gets freed. Other than using, we don't need to use a tracker, but also the thing is that my right hand now becomes easy for doing surgery, and it's available. Like you can see here, I'm using another drill with my right hand, whereas the left hand, the telescope is now navigating for me. This is an example of how it appears. Just to give you an example, like this is a case of the nasal cavity that you see, which does not have any known landmarks. So how would you know where the lacrimal sac is at this point of time? And this is where, once my endoscope is going, if you see on the left side, we are using something called as look ahead protocols. Now what it is doing is, there are 4 pictures where you see. This picture tells you where exactly your endoscope is at this point of time. This is 5 millimeter ahead of it. This is 10 millimeter ahead, and this is 15. So now what I have done is I have injected iohexol into the lacrimal sac, right? So you can see this as lacrimal sac brightening up. So now I know that it is lacrimal sac is in the same direction, is somewhere around 10 millimeters to 15 millimeters from the current location of my endoscopes. So this is where it helps you in localizing whatever your area of interest, anatomical interest is, live during the surgery. So CT will come live during the surgery at that time. Another example of, you see, I've come very close to the lacrimal sac. This is the peri-sac lacrimal vasculature, where it is showing me in coronal scan that within 5 millimeters from this point, you're going to touch the lacrimal sac. And by 10 millimeter, you'll be behind the lacrimal sac. So that is an advantage that this happens with this thing. So the advantage that this particular innovation gave us was of a continuous navigation during the surgery without having to use trackers. This is now published, and it gives the details of the maneuvers that we did. The second point is about balloon dacryoplasty. Now, if you use balloon, I won't go into the indications and where we use balloon dacryoplasty. It's quite commonly used with lacrimal surgeons. But the traditional lacrimal balloons, which are manufactured in the West, and now FCA is also manufacturing, on an average costs us around 26,000 per patient during the probing procedures. Now, how feasible it is for us to be using a good technology to add 25,000, basic 25,000 to it, even if we don't want to take extra out of it. What about the anesthesia? What about the procedure charges for probing and intubation and other thing? It was reaching around 40, 50,000, which according, as per the Indian standards is something which is, not acceptable. And that is where we thought, why not use coronary angioplasty balloons? Because coronary angioplasty, because of the volume, they're quite cheap. Like this one that is manufactured in Haryana it's approximately around 4,000 to 5,000 rupees per catheter. So we are reducing the cost by almost 80% to what is existing today. So we thought, why not let us experiment with these catheters and see. So this is a typical coronary angioplasty catheter. Obviously, it is very, very different from the traditional catheter, and you need to have a lot of modifications to be able to use that. Number one is it is very, very long because it's taken from the femoral root right up to the coronary root. So it needs to be there, quite big, like you can see over here. The second thing is that it has a very, very big guide wire before the pink balloon that you see, which we don't need because by the time we reach into the nasolacrimal duct, the balloon would not even have crossed the punctum. So we wouldn't like to do that one. And this is the lure lock mode for attaching to a manometer and an inflation device. So what we did was we started using them with the modifications and the techniques, the typical example clinically that you can see over here. And then we published and we looked at all the physical parameters and compared it with that and how advantageous can it be for the lacrimal surgeries. And we found, other than reducing the cost by almost 80%, we found that it actually does better on many physical and balloon parameters as compared to the traditional balloon dacryoplasty. This is also published, and one can have a look at the publication for details. The number 3 was redefining the anatomy and physiology of Horner's-Duverny muscle. Now this, I'm sure a lot of ophthalmologists know this muscle is Horner's muscle. We began by proposing, I won't go into the details, but we proposed that the name of the skeletal muscle should be changed to Horner's-Duverny. which has now been accepted by the anatomical societies across the globe. And subsequently, we looked at, the one important thing about anatomy is that we usually believe what is written in the textbooks. We don't want to go back and relook or redefine it and see whether what was said 20 years back, does it hold true today with the new technologies. So what we did was, this is a typical example of the canaliculi and the part of the lacrimal sac. You can see the common canaliculus over there. So what we did was that we took probably more than 10,000 sections of the single piece, all histological sections, 7 microns, every 7 microns we were taking one section, both upper, lower. And what we did with this is that we took it to Siemens in Erlangen in Germany. And Siemens does a very good stacking of histological sections to give us a 3-dimensional reconstruction. So we asked them, we gave this 10,000 sections to them, and we asked them to, can they use their own indigenous softwares, and can they rebuild this whole thing for us from internal, external, and everything. And this is how typically the software reconstructions look at different sections, both horizontally and longitudinally. And this is how it slowly then rebuilds image by image, studying all those 10,000 sections to build you the canaliculi and the lacrimal sac. So we wanted to study the Horner's muscle. So one we did was more than 10,000 per sample sections of these. And then similarly, we did many, many more. At every 1 micron, we took ultrastructural examination of all these tissues. Like you see this canaliculi with the surrounding muscles. I won't go into those details of how they look at it. But similarly, we put up all these images then together. And then we took it to pattern recognition labs, again in Germany, by a fantastic company called Chimera, which is just coming up. It's a startup kind of a thing, and they have developed fantastic tools for, again, reconstructions, 3-dimensional reconstructions using AI models. And then this is how we get the 3 dimensions of whatever samples we gave. And you can see this gray represents a lot of connective tissue, and this represents the muscle. This you can see the punctum. Now, what interesting feature, for example, one might say, so what? Like, for example, we ophthalmologists on the panel would know that we have been traditionally taught that there are a lot of muscles, the Horner's muscle is there, and there are muscles which are separate for the vertical canaliculus. We didn't find any such thing. We found that it's only a single muscle all through. And if you take sections now within the muscle, you can see that beautiful canaliculi going within the muscle. So this gives you a very different outlook using this technology to study the anatomy. And then you are able to go up to microscopic levels and again study anatomy 3-dimensionally. Like, for example, here I'm looking at the Horner's muscles, myofibers, and I'm able to measure all those things in 3 dimensions. And very surprisingly, we found that this skeletal muscle is not like any other skeletal muscle in the body, which was very surprising. And similarly, we could go and study the 3-dimensional anatomy of the innervating neurons by lighting them up with a lot of markers. We can do that one. We won't go into those details. So then you collect all these data and make sense of what is the anatomy. So unlike the previous belief of how the Horner's muscle, this is totally different now. Okay, so now this is how the arrangement is typically there, which is not there in the textbook. What was believed that it is longitudinal to the canaliculus is no more true. The anatomy is totally very different. And this is how it opens and closes canaliculi with each blink. And I won't go into those details. So, but what I mean to tell you here is that you have been able to now, with the use of these different technologies, you have been able to study anatomy totally in different light. You have been able to study innovations, and you have been able to study the biophysiology of it and come up with something very, very interesting. So this is in a little gist of it in a summary. So what I would go and end, and I suggest is that never give up. Believe in yourself, even if there is something which is sitting in Germany, some startup, if you can utilize their technology, how would that benefit lacrimal system or whichever areas you are working? Start looking at these things, and you can still start innovating without really having to wait for a device of your own. So thank you so much.
Thank you, Dr. Javed. And the beauty is you've actually covered almost all the questions that the audience have asked in your talk. So I've just got one question now pending, which I think we should ask Dr. Milind. What are the new insights into the lacrimal pump? Looking at all of this, is there what's the new thinking on this?
Well, Anand, the last part which Javed covered about the Horner's muscle and its changing anatomy. So the cases that we normally operate, they have an obvious block somewhere and we do a DCR and so forth. But functional epiphora is one big area where the system is patent and the person still has epiphora. And that is one area where the changing concepts of this anatomy, as well as how it actually in real time pushes the tears to the lacrimal system or does not push it so well, needs to be understood. So Javed is at the first stage where he has dissected the anatomical part of it. Now the next step there would be the functional studies, where initially I'm guessing it'll have to start with patients who've been operated for a DCR, where you make them blink and study internally what is changing in the lacrimal system, which wall is moving and so forth. So the next step would be dynamic studies, either markers within your tears and how fast they are reaching down, as well as endoscopic live videoscopy to see what's really happening, whether that anatomy is reflected in the functionality. That's my opinion.
Thank you so much, Dr. Javed. That was beautifully illustrated. And congratulations on the pioneering work and all your achievements and awards. And may you go to greater heights from here.
Thank you.
Our next speaker is Dr. Anand Shivaraman. He is really somebody who brought back a lot of knowledge from the West and then started his own company here. He's master's, PhD, and postdoc from MIT. But then he came back to India because he wanted to firsthand experience this the knowledge that he had, and to make it more accessible and all what he is doing much more affordable to reach the community. He's going to tell us about his journey. Over to you, Anand.
Thank you so much, Dr. Anand Vanekar and Dr. Chaitra. I think I cringed a little bit, and I'm sure my mom would have also cringed when Dr. Vanekar called me Dr. Shivaraman, as I think we are the PhD kind that may not be very useful when it comes to clinical. But moving ahead, I think one disclosure in terms of conflict of interest is I'm part of Remedio, CEO and the co-founder of the company. So just to make sure that things are also brought in perspective, there is a background to how Remedio got started. And I think it is no less similar to a Bollywood script, where I think Indian Railways is someone that I would owe a lot of thanks to for us getting into ophthalmology. The Jab We Met moment, I guess, for us and Remedio was when we met Dr. Anand Vanekar on a train journey from Bangalore to Cochin. At that time, I was an engineer dabbling in a lot of optics and in vitro diagnostics. And I think the only difference, I guess, is Kareena Kapoor was replaced by Dr. Anand Vanekar with the same amount of exuberance and the impact that he had on us. And the rest, I guess, is us getting into it in 2009 and starting to ask the question how we could have impact. So the script actually unfolded where we initially started working to see whether we could build affordable ROP devices. But what was very interesting is there was the time when we started building the first prototypes and then miniaturized it and realized that it could actually have a role in adult diabetic retinopathy screening, and that's how the Fundazon phone came about. So it was completely a classic case where you ended up starting on to make something else, ended up making something totally different, but then realized that there is significant impact that can be had as well as commercial value. So that's really how the script really unfolded. So the only message is let's all be open to the fact that accidents are wonderful things to happen as well. What I wanted to quickly focus on is to answer some of the questions around the culture of innovation, which we really want to build grounded on good science in India. And I like to use this particular slide, which is if one looks at how technology obsolescence is happening, you possibly have within 15 years technology actually getting obsoleted. And oftentimes for companies like, let's say, Apple and Samsung, the reason they have been able to continue on in spite of not getting disrupted is because of the continuous innovation that has kept them relevant. So one of the things that we've tried to do, keeping this in mind within Remedio, is to look at the fact that, is it possible for us to operate on the next curve? Can we think in terms of technology that may be at the boundaries today, rather than necessarily only technology that can be made primarily from the point of view of affordability? This is something that we believe strongly in, because of the fact that we think that making in India will naturally make many of these devices affordable, especially when we are able to match that with global quality standards. So some of what we've done, therefore, over the years have been primarily starting off with in 2013, where we were one of the first to do the Mediatic imaging on a smartphone, to, I think, where we possibly are still today the only ones doing non-Mediatic on a smartphone, to a variety of solutions, all of which have come about as a result of us looking at the needs statement. And I guess one advantage we have is, since we come from a very different background, we tend to therefore question the status quo and ask, is something can be done differently? So we've had anti-imaging modules that can convert slit lamps into digital slit lamps. We then brought in offline AI as a means to be able to help in public health screening, to microscope recording devices. This particular example of C3FA was a case where a young innovator, Sandal, was working with Arvind Eye Hospital, and it also formed a template for us where we realized that there is a lot that we can do in terms of working with the ecosystem, especially if we are able to ensure that we can help them in their journey. And that's one example of how we did it. And I just wanted to also bring out the fact that sometimes it's always helpful where the government is actually providing a large number of grants as a means to be able to bring some of the early technologies to commercial relevance. And those are available, and therefore to all ophthalmologists and all innovators. One of the points I would make is more than happy to work with everyone, and we find out first if there may be readiness to the product in spite of a patent or otherwise, and look at being able to take a number of grants to advance them to a point where they may be ready commercially. So we've also, for example, this year launched an AI and infrared enabled portable slit lamp as a means to be able to do screening. And one of the new devices or new technologies we're working on is to ask the question, as a result of us having worked on being able to miniaturize many of the optical designs, we asked the question, any reason why in an ophthalmologist clinic you have 4 different rooms having 8 different devices? Is it possible for us to bring them all together in a single automated device? This is something that we are working very closely with an Israeli company, where we have both shared our patents on the imaging and the aberrometry side. And that's an automated 8-in-1 device that we're working on. The key message I do want to give is, I think all along in India, we've focused on affordability. We believe that affordability can be had in India, but the step to challenge ourselves as an ecosystem is, can we actually operate on the next curve? Let's make in India, but attempt to be the world's first in whatever we create. And that is something that I think I believe very strongly in, and I am extremely positive about that being possible in this ecosystem. The other is let's patent and or publish. The reason I say and or is depending upon our beliefs. There may be a lot, many folks who may not want to patent, but I would strongly encourage all of us to publish, because it's a global currency for credibility. And we've had a number of publications that has actually helped us believe in the science that we have built the technologies on, and also at the same time have helped us with credibility when we actually are taking these devices global. So that's the second message that I want to leave, where we, both industry as well as the clinical ecosystem, our ability to publish as a result of the advantages we have with the clinical cohorts in India would be something that would be truly fascinating for us as India to bring to the world. We've had more than 2,000 plus installations of the different devices, and many in the US as well. And the reason I find this is very interesting, and I want to just spend 5 seconds on, is a lot of what we learned by working in the field in, let's say, hub and spoke models, where we were in the spoke in vision centers at Arvind, or with Dr. Mohan's robotic centers in spokes, where you may not have those that are well trained to use the device. served us very well because the same devices are being used in primary care in the US by a nurse practitioner who has possibly equal or lesser knowledge as a specialist would. And therefore, the idea of being able to build something very simple in India and take it global, I believe, is definitely a model well worth thinking about. I just want to quickly segue into the devices with integrated AI, which is the next frontier that we believe would be very interesting to look at. Some of the possibilities with artificial intelligence enabled devices, some of which I think Dr. Kim and Casey have already talked about. One that we know very well is the use of automated algorithms in public health screening to identify, let's say, those in need of a specialist, sort of what we call triaging. And in that, we've been working on DR screening. We have a commercialized product that is CE marked and been brought to market in India. And we are simultaneously also working with a number of groups, most notably with Narayan Netralia, in looking at glaucoma, ROP, as other examples of where we may be able to bring in AI to help with automated diagnosis. At the same time, one of the things that we've learned as a hardware company is AI is not only about automated algorithms. It is actually helping us to build some very intuitive devices by assisting a health worker or a technician. For example, we've taught the AI how to do auto capture, autofocus by training them on images. In fact, there was one very interesting attempt that we did where we had certain glaucoma specialists who were looking at the pallor on a Zeiss hardware system versus that coming from ours. And we actually taught the AI to convert our images to look at the same pallor as what the Zeiss images would look like for standardization. So there is a lot that is possible with AI, which is beyond just automated diagnosis, even as I think we need to make sure that there is effective prospective, cross-sectional, as well as retrospective training and validation that is made possible for the AI. One of the new areas that we have also started looking at with AI is, can we enable new insights through multifunction devices, especially in the effect of treatment over time through longitudinal parameters that are available, and also start looking at what we call eye wellness scores in screening. Because at the end of the day, while we start with diabetic retinopathy screening, it is about going beyond that into refraction as well as other retinal diseases as well as glaucoma. So in developing an AI algorithm, it is a trial and error process. Data is the most important part of it. And one of the beautiful things in India is the ability for us to have a very diverse clinical cohort that is able to give us the kind of quality of data that is needed in order to select that and use it for training. However, at the same time, there are certain challenges too. For example, when we built our DR algorithm, we realized that the ground truth acceptance for mild NPDR as well as severe NPDR across multiple graders was not very high. The kappa values were not very high. So we had to therefore choose ways by which we develop the algorithm so that it's not affected by constraints as a result of a lack of agreement across graders. But to a large extent, these are the typical steps involved in building an AI algorithm, which is data selection, training of the model, validation, preferably prospective validations, and eventually we monitor the models post-deployment. This is true for any of the AI algorithms that are developed. So some of what we've already brought to market is one on referable DR, and we initially worked with a group called INAC, and because of the challenges that we saw with using online AI where it needs infrastructure such as constant internet access, we started working on developing what we call offline AI, which works on the smartphone itself, where within 6 seconds, it automatically finds the areas that may have telltale signs of referable DR and is able to say whether or not there is a need for referral. We are now working with Narayan Netralia and other centers for us to start looking at validating sight-threatening DR algorithms as well as glaucoma algorithms, as well as on retinopathy of prematurity, where we are looking at being able to launch a smartphone-based large field of view device to be embedded with the AI algorithms for screening. So these are some of what we've been focused on, public health screening using AI. While at the same time, one of the frontiers that we're looking at is the advantages of offline AI for us is the fact that we don't need to depend, especially in a developing world context, on infrastructure. And for me, this was very poignant because I do remember that in both Armenia and in India, we had certain screening models where the patient would ask us, why do you call us for getting screened if you're not able to instantaneously tell us whether we need attention? So that's really when we got into being able to build an AI which is integrated into a device and which is able to give an instancy in terms of the way it actually comes up with its own automated analysis. At the same time, all of this has to go through current accepted global standards for sensitivity and specificity. So we use the FDA and the MDI Europa standards in the absence of standards in India specifically for this. Offline and on-the-edge deployment of AI also ensures adherence to strong privacy requirements that are especially needed in the EU market. And in the developing world context, infrastructure is then not a limitation as a result of the use of offline AI. So to a certain extent, we like to think of it as how do you design for constraints and then take it global, which is really what developing in India allows us to do. At the same time, this is an example of where we are doing some new research where we are building retinal cameras that need absolutely no infrared image. We realize that the AI can automatically identify when you are at the right distance and automatically click. So some of the work that we are doing is to make the devices simpler as a result of eliminating some of the traditional methods by which imaging was done. This is something that we are getting into and also answers a question that was posed by Dr. Ravindra, which is we're starting to now look beyond just the case of diabetic retinopathy, but are starting to look at training images and training an AI with images from cataract as per LOCS scale and start looking at being able to see whether or not we are able to predict staging. Of course, AI works very well in those cases where there is a certain standardization of clinical result. And that's where we've gotten into, after diabetic retinopathy, into cataract. We are also getting into starting to look at glaucoma from the point of view of both angle closure as well as open angle glaucoma, as well as topography and keratoconus. At this time, it's very early. We are developing, we have just completed the development of the system and are starting to collect data. Now, with multifunctional data of the sort, the opportunity is the fact that you're no longer looking at just one particular result from the eye. The way to look at it is when you actually have blood work that is done, you have multiple markers that are identified from the same drop of blood. So there's no reason why we can't therefore think in terms of technology that gives multiple outputs from a single consult. So this, for example, within 2 minutes completes the ARK, Scheimpflug, fundus imaging, as well as retroillumination for cataract and topography, and also has a dry eye module in it. So these are early days, but I'm extremely excited because with more data, AI then has the ability to assist a doctor far more and also make themselves available in looking at those that may be referred from public health screening. So these are going to have an impact, at least in our opinion, in public health screening, as well as at the same time in ophthalmology. Of course, at the end of the day, as an entrepreneur, it's also about how do you bring something like this to market when you know that there may be additional efforts at being able to make this as affordable. And that's where I think the advantage of being able to manufacture in India and bring these to market actually make it extremely exciting for us. So just want to end by saying that it's a gratitude that each of us have to this ecosystem, because I think we've learned a lot from every ophthalmologist, optometrist, and biomedical engineer in India. As Casey said, many of us who got into the space, I'm a chemical engineer by training. And so at the end of the day, a lot of what we've learned is as a result of what we have learned from all of you in the ecosystem, and specifically a few of the institutions that have worked very closely with us and we are highly indebted to. And one of the things that I did want to share is we do have a reasonably global workforce, and that's helped us a lot. A lot of the AI that we are developing is in Singapore with our subsidiary company. And a lot of the exchange of ideas, especially when it's cross-functional, is resulting in a significant number of innovations that we think is extremely useful for us from an ecosystem perspective. So I'll leave it at that and happy to take any questions.
Thank you so much, Anand, and for jogging back memory of that Jab We Met moment. You've really gone a long way in this road, and so happy to see all the progress that you and Remedio are making. You also answered a lot of questions related to AI and glaucoma and cataract, so I won't ask you that again. But I'm going to ask you a super difficult question. What is the one technology or product that is not yet available globally that can significantly impact the field of ophthalmology? Not available.
So I thought, Dr. Anand, I gave that question to ask the rest of the panel, but I realized that the question coming back to me.
Well, let's get your wish list.
Sure. I mean, I would love to hear from the ophthalmologists in terms of what that one technology would be. The reason to put that question out is while we are going the journey of multifunction and giving more information to the ophthalmologist to better manage a patient, that's been our approach, and also bring in AI as a means to triage and make it easier for the ophthalmologist. I would love to hear from the rest of the panelists on whether they may be able to think of some technologies that today don't exist but can significantly disrupt ophthalmology. And we have, of course, only covered on the OPD and the diagnostic side, really not on the surgery side as much as the examples we've given.
Go ahead. If any of the panelists want to throw in their wish list, this is the time. Where do you see? Yes,
I personally feel like in terms of ophthalmology, one thing which AI will really contribute, like everyone's mentioning, is helping the ophthalmologists in entering the EMR. I think that's one very important factor. This right now, I mean, oral dictation is something different, but the connection of the different devices into your findings directly coming into your slit lamp. In the sense, a photo is taken and all the findings directly fills in the space of your slit lamp findings. And also, let's say if you're taking an OCT, a series of OCT, or let's say it's a follow-up OCT of a patient, and immediately your EMR is already saying, okay, there's amount 20 millimeters of difference in macular thickness, or whatever, if you're talking about macular edema and so on and so forth. So this, definitely AI is not a threat to our jobs, but definitely it'll make our lives more simpler, and it'll make our job, we can do our job much more efficiently. Yeah.
Thank you so much. Chaitra, you can take it forward.
Yeah, so our next speaker, I'd like to introduce Divyansh. He's currently working as a consultant with retina and ocular oncology at Sankarai Hospital in Bangalore. He's been there since 2013. He's received 7 prestigious awards in various state and national forums. The most recently being the Colonel Rangachari Award Overall Best Paper at AIOC 2018. Dr. S. Natarajan Best Paper in Vitreoretina in 2018 as well. Dr. Mahashebe Award for Community, and Dr. Bashir Naikri in KOS 2017. Thank you, Divyansh, for being a part of this, and over to you.
Thank you, madam. Thank you, Anand sir, for giving the opportunity. It's an honor to share the dais with mentors of my mentor, Dr. T.P. Das and Dr. LG. It's really an honor. And thank you for giving us the opportunity to share our experiences and how we had come out with the innovations. So I would be sharing my screen. Is the screen visible? I am audible?
Yes, both. You can continue.
Yes.
So good evening, one and all. So the topic which was given by Dr. Chaitra madam was that young innovators. I would think probably innovation would not be an idea, because as the definition goes, the process of translating an idea or an invention into a good or service that creates a value for which the customer will pay, I haven't been to that. So I would dodge this and then probably opt for something which is more apt for us or me, something which is called the frugal innovation, or a jugaad in Indian terms or in Hindi word. What does frugal innovation means is to strive or create goods or quality solutions in order to deliver great value to customers at low cost. And most importantly, in a short sentence, if we have to form it, ability to do more with less. And what all restrictions do we have in a frugal innovation is, as we know, there are 3 main restrictions. Yes, affordability is the biggest constraint, because the place where we work is in is an NGO. And again, the other thing is the institutional complexities, where yes, we need to work within that. And most important, the resources are also limited. I would like to thank all my contributors which are there in this photograph and which were there in past and right now. And yes, not to mention or not to forget to mention my head of the department, my mentor, Dr. Mahesh. So what does innovation require? It requires three important things. One is curiosity, mentor, very important, and again, mentoring by a mentor, and an environment for the innovation. So as you can see, this is my child. This was the first instance where we had took him to a mall to try the scooter, and then he wanted to jump on too. Why this? This is just a curiosity. And as we know, at no time in life is curiosity more powerful than in early childhood. And as we grow older, the curiosity decreases. I don't know what the reason is, but yes, if the curiosity survives, then we can innovate. And yes, the most important, or the second most important, would be the mentor or the mentoring. I would like to thank again my mentor. Yes, the training is very important. What to do, how to do. Keep motivated, even if you have not achieved in the first instance, probably the second instance, probably the third instance. Yes, advices on each and every positive and negative instances to how to manage it. Success, yes, take it with a pinch of salt and then go in a correct direction and again try to innovate further. Coaching, yes, as we were discussing, it includes all mentoring with the coaching and support, yes, which he always gives to us. And we have to work towards a goal. The third most important thing is the environment for innovation, where you have to create a role model, like you have to become Shah Rukh Khan, or I wanted to become my mentor, or wanted to at least try to replicate or implicate what my mentor has done. So that was my role model, and to have some recognition by our colleagues, seniors, that you are doing good work, that gives you a big morale to work for it. And yes, to work in a safe environment and explore it, provided the time and resources. Very important. If I am not in the OPD doing some other work, somebody has to work for me. So again, thank my colleague, Dr. Rajesh, because most of the times he had to bear with the OPD once if I am not there. Always search for the second right answer. So there is not only one single best answer, it's a second right answer probably which would help you to go innovate and makes goals and strategies. And yes, after all this, you may have a eureka moment that you have achieved. In our childhood, everybody would have a role model as our father, and as you grow, it may be probably an actor, an actress, or something. But yes, for us, once we are in a fellowship or probably in our career, our mentor would be our role model. So this was a paper which we tried to implicate, as he has used a handheld video camera. We utilized the phone, which was very easily available. But yes, the most difficult part of it was to hold it. And yes, as you can see, many reflexes and issues that how it has to be held and ambidextrosity, which was utilized. So we thought of the much easier option, which was a direct ophthalmoscope. Yes, there are many negatives that it can only see 5 or 10 degrees. You have to go close to the eye and things like that. But yes, the very important thing was that it works on basic principles of light and reflection, that either we need a partly reflective mirror or either a hole within that mirror or probably a partial mirror. And the upper part of it we can use. So this is what we thought, that yes, how we can do that. The third part can be utilized where only the part of the mirror is there, where the light is projected into the eye and reflected back into the camera. But in a smartphone, this is my oldest smartphone, which I got it out today. And then this was a smartphone which we started this innovation. As you can see, that actually, if we have this light very close to the centermost point of the camera, it would work exactly as a direct ophthalmoscope and nothing needs to be done. No lens, nothing at all. So this is what the clear optics was, that we need to get it close. Okay, so how do we get it? As you can see, it's a real muck. So this was the first and foremost trial which we are doing, where as you can see in a diagram, we have passed some 3 fiber optic cables and we have used 2 kind of lids, which were the beaded bottle lids, and then we have stuck some kind of a tape. And these are the 3 light optic cables which once goes onto the light gets illuminated. As you can see, it was not working well, and probably there are some multiple jutting fiber optics. So this was a more refined work. As you can see, we got an acrylic sheet where we passed 3 fibers, and then this is the side which would go onto the phone or the camera, and then these are the lights which would, or the fiber optics which would come out. And how would that function? As you can see, once this fiber optic is over the light, then it gets illuminated, and not on it, then it doesn't. But it helps us to push the light more closer to the center of the camera. So that was our achievement, but yes, it was not as good. This one was very bad. My wife was not at all happy because I took one of her clothes which were having this reflective mirrors, because we thought probably we make some kind of a box where there are two reflecting mirrors and then help reflect the light so that all this jetting process would come out, and then it would be much more compact kind of a contraption. But yes, this didn't work and my wife was not at all happy. This was some kind of a diagram which we thought at that time that there would be two rotating mirrors which would reflect the light from one, and then the other way it would come out. Yes, this was very important, which we tried was the 20 diopter lens, the glasses which people wear, we cut that into kind of a rhomboid where one end was made with a silver. So this is my wife's silver paint, as you can see, and then we have covered it with the tape so that the reflection occurs correctly. As I would again play the videos, as you can see, the light is reflected from its particular area to a more towards the camera. So that is what we wanted. But yes, this was more diffuse. As you can see, there are a lot of reflexes and diffuseness. So this was kind of a ray diagram which we thought that the light would get start from here and then get reflected with the mirror over here, and then here it would come out. It was working, but still not as good as what we wanted. Then we thought, then why not to keep a LED and then finish the job? And this was our first publication in 2014 where we did that. And as you can see, some areas of fundus was seen, not though very clear, but yes, the important point was that it was still jutting out. So what to do next? Then when I was driving back with one of work, and then I saw that some LED lights are glowing, and those are very flat lights, and those would not have this jutting problem and would be very focused light. So we have covered other two LED lights, and the third is open, and this is powered by one of the batteries. For an entry segment, either you can use a 20 diopter lens over here, the intraocular lens, or our LIO lens, and as you can see, the images would be very good to take either in an OPD or an examination, because our institute do not own a red cam or a kind of imaging device in pediatric cases where it requires in an OP. So this is what the image is, as we can see. So this is the same contraption which we have arranged, and then we can indent and even see the periphery. Yes, this is a direct ophthalmoscope on a smartphone where you indent and you can still see the periphery because you can tilt it so well. And then you can see the quality so well. You can see each and every blood vessels, the yellow tinge of the macula, and the cystoid changes of the tumor itself. But yes, the wide field view was still not there, as you can see. So this is a comparative image. I hope that everybody would be happy with this image. This is a comparative Topcon T40, and then this is our contraption which we had made. Yes, it works well. Does it work well in an uncontrolled environment? So we did a study of around 200 patients of diabetes, which included cataracts, pseudophakia, and others. And as we can see, we had again compared with commercially available devices, which are DI and the peak retina. And this study was done in collaboration with Bonn University in Germany. The study was done in India. As you can see, the images are very well over here. This was a similar kind of contraption which we are thinking that the light should be reflected, was utilized by the DI. And then again, some kind of an LED around the camera, which is what is in the peak. But as you can see, the reflections are gross. And in R, where the light is very focused, the reflections are not much. So in our study, the DIY seems to do well. Yes, the learning in this innovation was, yes, the curiosity has to be there to do it. There has to be right conditions and right mentor to help you. And there is an iterative up process. As you can see, there are multiple failures which we went through, but yes, we have to continue. So this is what is called as idea funnel, which has to be continued, that we need to come up with new ideas and new idea in pipeline, and the velocity that the next idea should come up very fast, and that is what is called as an improving batting average. So to increase your response rate, also to try other things. But yes, the most important challenge we need to face is overcoming the resistance that yes, it may fail, and probably prediction of disability. But yes, this is again a big question, whether we publish or whether we patent. We didn't patent this. Yes, we tried to publish all. That would be my question to the panelists later in my presentation. So we went on to the next thing for a wide angle imaging system, which again, as I told, that our institute doesn't have either 3-netra, Netra Neo, or either red cam. So we utilized a nasal endoscope, which was put on to the camera of the smartphone, as you can see, with a small, very easily available adapter. And yes, these were the images what you can see. The quality is a little subdued, I would say, but yes, all the details which you want to see, the disc, the traction little detachment over here, the kind of calcium which is showing a lot of reflexes, small areas of probably inactivated tumor, RP atrophy, as you can see very well seen over here. And these were the images with a handheld camera. So the handheld camera, we need to take multiple images, but if you have a single wide field image, that would be very good. As you can see, yes, it can be utilized for anti-segment imaging also. Very clearly, we can see all the angle structures as what you can see. So the wide field OCT, which we got the best paper in AIOS, is we utilized again the curiosity that whether we can do anything better with what we have. We had a 12 mm scan, so we tried to do something better. We added a 20 diopter lens. Then sir gave an idea that, okay, try something else. So we tried pan fundoscope lens and then other lenses. But yes, the best which worked was the 78 diopter or a 90 diopter lens, which gave us widest field of view, which was around 30 or 31 mm. Okay, whether does the OCT functions well or whether the opta can also be done. So as you can see, yes, the opta can also be done. As you can see, there are a lot of artifacts which are there in the later part of the image, not in the initial part, is because of the tiredness of the patient not able to focus. Yes, but the widest field of view of opta, which is still not available commercially, can be done, which is 20 by 20. And this is how it can be done, that you make a patient wear a trial frame and then put a 20 diopter lens and then do it. Or either you can hold a 90 or 78 diopter lens and then do it. Ideally, a manual arrangement has to be done to change the focusing and fixation, and then you would be able to get a better scan. So this was one of the collaborations which we did, where we found out a transpalpebral or a transscleral illumination, which gave us such a wide image. And this is, mind you, it's not a dilated image, it's an undilated image, and it is actually comparable to a Topcon fundus camera. Yes, we can go till around 120. There are pros and cons associated with the transscleral or transpalpebral imaging. As you can see, the hotspots can come up, but yes, all imaging devices have some pros and cons associated with it. Yes, we had some stint with having a collaboration with the Netra AI, where we did our collaboration and did diabetic retinopathy in AI-based algorithm, which had a very high sensitivity and specificity, somewhere around 98-99%. Again, this is a very important video which I wanted to show that yes, innovations can not only be in your imaging, but can also be in your OPD. As you can see, this patient is unable to read beyond certain lines.
Here, you have to chin up and read it. Just keep it one second.
I'm not sure the video is probably stuck for me.
Now.
So I just go back to this again. So as you can see, so now she is reading with one of the lenses in front of the eye. It is called polarized lens. So once we keep that lens, the vision improves. So actually from 636 or 660, the vision is improving to 660. What this does is cut the stray light, and only a light, a part of a column of light which is passing through the filter is allowed inside, and that's how the patient reads better. So this we found out that it is helpful in case of macular degeneration, optic atrophy, and various other diseases. As you can see, the speed of reading is also very fast. Yes, there are some COVID era innovations. Most of us would have seen that in patients, if the mask is not tightly fitting, then probably the objective lenses of the OCT may get affected or get fomites onto it. So a simple Kringlac, I'm sorry, the video is not functioning. So a simple cling wrap can be put onto it and then probably use isopropyl alcohol to wipe it. The quality of images are very good. As you can see, only the flash reflex which you can see in the center, but otherwise it's very good. Yes, something in kind of a treatment. This was the idea by Dr. Mahesh sir, which we used a 577 single bandpass filter to treat recalcitrant cases of macular edema, which have taken probably 10 injections, 15 injections, and they're really frustrated that we do not want to invest anything more. So these cases also did well. This was a surgical innovation which we thought of. I being not learned in an era where buckling is not so commonly done, for me to identify a break or to see a localization of a break or to see whether it is nasal, temporal, it was very difficult. So a simple bent illuminator can be utilized to identify the breaks or identify where exactly the buckle is, whether it is posterior to the buckle or anterior to the buckle or break. And you can mark the posterior most edge, the lateral and the nasal edge. And if the cryo is adequately done or not, it can be attached to the cryo, and then we can see that whether cryo is also adequately done or not. So this avoids the confusion of the inversion and also the lateral inversion, which occurs whether the image is in the nasal part of it or the temporal part of it when we are doing a cryo or when we are identifying whether the buccal is adequately covering or not. So this was again a very innovative idea by my mentor Mahesh sir, that to utilize a 100 rupee note where the numbers which are there over here, we identified that at 2 meters, it subtends an angle of around 1 minute of arc, which is approximately 612. And we found out that 612 was good enough to identify children who have a vision abnormality which can be referable or non-referable. So when we were doing this kind of a test, this one of the child, I will show you the video, probably you may hear it out, that this child is doing the similar test once she is going home. So as what she's talking is that probably you are not able to see the lower numbers, you need to go to get an eye checkup done because your vision is not good, or either it's a referable kind of a vision loss. So again, a simple 100 rupee note itself can be utilized where the numbers can be kept at 2 meters and the patient can be asked. Yes, the illumination probably using a daylight would be better. In the night, it would not be possible. So some myths and realities of innovation. Does innovation begin with creativity? No, it begins with the curiosity. To look a question or to find a question in the simplest of things. And whether the innovation is a smooth linear process? No. As again, I had shown that it has to be iterated and the outcome of a series of experiments which can be either failure or either positives. Innovation is about risk-taking? No. It is about risk-taking and assessment and mitigation, that how much it would have an effect or pros or cons of each and all. So this would be my questions for the panelists, that whether to publish once you have an idea and once you have already validated it, or to try to file a patent. As you know, once you have filed a patent, it would be very less amount of collaborators who would collaborate with you. As again, Dr. Subain sir told that probably people would not accept that you have already patented it and they would not be able to collaborate with you. And to decide on how to decide that ideas or innovation needs an IP protection, and how to go about to find a collaborator to get it to the next level or the product level. So again, thank you all, and I would like to thank all my team members, my mentor, my colleague, all and everybody at Shankara. And thank you, madam, for giving us the opportunity. Thank you.
Thank you so much, Divyansh, and congratulations to you, Dr. Mahesh sir, and the entire team at Shankara. And you may actually made my life simple by asking those questions, you know, and you actually answered all the other questions that the audience has asked. We will reserve the question taking for another 2 more, because that's got to do with lab integration and taking but you've asked those questions, and I think it's giving enough time for the panel to think about it. So let's quickly move on to our next speaker, Dr. Arka Ghosh. Dr. Arka heads the lab, the Grow Lab at Narayana Nethralaya, Bangalore, and he's going to talk about integrating lab and the clinic.
Can you see the screen?
Yes, Arka, you can continue.
All right, good evening, everyone. And thanks, Anand, for the introduction. So today I'll explain to you how the laboratory gets connected to the clinics. I mean, we've been talking about innovations and how to bring newer technologies, newer knowledge into the clinics. So I'm going to take a few examples and show you how we've done it using the Grow Lab example of systems medicine. So the idea of systems medicine is that you take clinical analysis, clinical data, and clinical samples. And these samples, you do in-depth analysis using a variety of different omics tools, a variety of different models that you can generate, and put these together into a matrix where you can statistically analyze the newer imaging data, the clinical data, the phenotypic data that comes from the patient, and the notes of the doctor, and all of this molecular data that comes from the laboratory, and put it together to build a story, to build a network that explains what is going on with the patient during that time in that disease. So using that information, you go on and develop a variety of different solutions for that. They could be for diagnostics, for prognostication, for monitoring, for new therapeutic targets, if there's an unmet need. So these essentially form the connection between the laboratory and the clinic. So what we are trying to do now is to develop new strategies for intervention, to do targeted therapies, personalized medicine, as well as develop point-of-care diagnostic kits. So at Aurolab, we work on a variety of different conditions across the different sections of the eye, be it the ocular surface, retina, glaucoma, etc. But what really is the strength, what you need to have when you want to do systems medicine is the depth of analytical techniques. So which means you should be able to do both genomics or proteomics, as well as transcriptomics, as well as AI-based statistics, develop various models in vitro and in vivo in which you can test your hypotheses, and also the platforms for coming up with novel therapeutics. So all of these put together gives you the depth necessary to bring an innovation to the clinic. So this is an example of how we do biomarker discovery work, and my colleague Swaminathan will explain more about it. But essentially, the story starts at the clinic. Your clinical partner is responsible for collecting these samples that you will actually analyze. So you all have to be on the same page in order for these things to happen. And what this knowledge of molecular pathways and molecular analysis gives you is the pathobiology of the disease. It can lead you to druggable new targets. It can lead you to monitoring of disease so that you can have better patient responses, so on and so forth. So I'll take a few examples and show you what I mean by this. So let's take diabetic retinopathy. So here, what we've done is we've taken diabetic retinopathy patients, and these patients are segregated into controls, which is the gray bars, then the treatment naive, which are these orange bars, the responders to treatment. So these are the green bars, the non-responders are in the red bars. So what you can clearly see is that when you take IL-6 and IL-8, two well-known inflammatory cytokines, they are extremely high only in the non-responders. So there's a story that's in there that's telling you that these patients who are not responding, they're likely not responding because they have higher amounts of inflammatory factors and not necessarily VEGF. So what does inflammation do to cells? So this is a movie to explain, to show you physically how inflammation actually influences the cell. So on the right side of your screen, you can see these cells. So these are epithelial cells. And what you can see inside these cells, this is the nucleus, and these are all the intracellular organelles. These cells are sitting around quietly, and they're just, the organelles are moving around. Now, see what happens to the same cells when you add an inflammatory factor to it. So it is TNF-alpha stimulated. These cells are running all over the place. They are gliding over each other. They have become hypermotile, and they are secreting a lot of stuff. So this is physically what's happening to the cells in your retina, in your cornea, and anywhere else in your body. And what do we learn from these things? What can we do with this knowledge? So these graphs show you VEGF and IL-6 in diabetic retinopathy, and as expected in case of severe NPDR and PDR, you have higher levels of VEGF and IL-6. So what happens to cells when you have all of these factors higher? So take a look at the VEGF receptor. So these black little dots that you see, they represent the physical VEGF receptor on epithelial cells. Now look at the red boxes. So when you add VEGF to it, the VEGF receptor goes down because VEGF binds to its receptor. The entire receptor and the VEGF goes into the cell to do further signaling, and it does not respond to any more VEGF. But now, if you have hyperglycemia, you can see that the VEGF receptor is no longer going in, even after stimulation with VEGF. What does this mean? This means that the chronic inflammation because of underlying hyperglycemia caused a maladaptation of a protective signaling. Therefore, that is the cause of your damage. So could this be a new therapeutic strategy? Possibly. So what the laboratory gives you are answers to important clinical questions. That why is VEGF signaling off in diabetic retinopathy when it is normal in other, if you don't have hyperglycemia? So a variety of such questions can be answered. So we take another example here. This is keratoconus, and we showed that in keratoconus tears, you have very high levels of MMP9, which is an inflammatory molecule, and IL-6 as well. But concurrently, what happens is you have reduced amounts of lysyl oxidase and collagen 4, etc. So you can see this here in keratoconus patients. You can't see any of the lysyl oxidase, where in the donor healthy controls you can see it. So what does lysyl oxidase do? So in case of the cornea, the lysyl oxidase binds together parallel strands of collagen. So if you don't have this binding done, these collagen strands get loose and they are slipping over each other. So if inflammation is high, can we reduce inflammation and reduce the effects, the pathology of keratoconus? So we did a small clinical trial repurposing cyclosporine, which is already approved for dry eye disease. And we found that that is indeed the case. If you reduce the MMP9 levels in a subset of patients, not in all patients, but in a subset of patients, it seems to stabilize the disease. Similarly, the same lysyl oxidase, how does it apply to monitoring or patient stratification? So in another study, we looked at patients undergoing cross-linking, keratoconus patients undergoing cross-linking. And it turns out that if your cross-linking results are suboptimal, in those particular cases, the lysyl oxidase levels were significantly lower than the cases that had optimal cross-linking results, which tells you that even for a surgical kind of solution, the molecular factors play an important role in their success. Taking the case of dry eye disease, what causes ocular surface discomfort? You have high levels of inflammation, you have MMP9, and what we recently showed is that there is a disbalance between the pro-nociceptive and anti-nociceptive factors. And this, herein lies another opportunity for us to intervene. What we did as a novel approach to handling this is looking at autophagy, the cell's system of removing garbage. So if you look at this image here, so when you give desiccation to epithelial cells, you see this color change. And when you add trehalose, which is a sugar, this goes away, because in during this desiccation stress, there is a disbalance between the ions in the inside the cell, and that is one of the root causes of the disease. So trehalose came out as an eye drop, and we did a small trial to show that it does work in dry eye patients, and it's now available. And this is an example of a partnership between a pharma company and us and a laboratory to bring out a product in the market. So the laboratory gives you novel solutions. In the final example, I'll take the example of retinal dystrophies. So retinal dystrophies, as you all know, is inherited in most of the cases. It's progressive, you lose vision, and currently there's very few solutions for these. In our hospital, we do genetic testing for it. But apart from the testing, we also want to provide a therapy, a solution. So therefore, we are developing gene therapy in our Iterative. So in order to do that, we have to make the drug. So this is where infrastructure and technology must come also within the ophthalmic workflow so that you're able to do the things that you want to do. So of course, this takes a lot of investment and a lot of time and a lot of technology development, which has been done in Iterative so that we can bring gene therapy into India. So the laboratory therefore also helps you develop innovative new paradigm. So in this case, this is personalized medicine that we are developing. So with that, I'd like to thank our clinical team, our Aurolab's team, my collaborators, and of course the funding. So this is, we discussed this, other speakers have discussed this about how can you bring an idea to fruition. You can't do that without funding. And I must say that apart from the generous funding that I receive from Dr. Pujan Shetty and the entire NN management, we have a lot of funding from various federal agencies, but a lot of funding also comes from a variety of industries. So I've listed some of these people here, and I'm quite happy that even Indian industry always comes forward to support innovative new work. So with that, I'll take questions if any. Thank you.
Thanks, Harko, for that excellent talk as usual. I think we will take questions together after Abhijit finishes as well.
Sure.
So now to introduce Abhijit, he's the Director of Imaging, Biomechanics and Mathematical Solutions, Navigational. He leads advanced imaging initiatives related to tomography and to biophysics, and he has over 100 publications actively pursuing to translate technologies being developed at NN. Over to you, Abhijit.
Thank you, Chaitra. I'll just share my screen. All right. So first of all, thanks for the introduction. So today I'm going to talk specifically about an imaging innovation that we have been doing at NN for the last couple of years. And this imaging innovation again deals with OCT. And I won't go into the basics of OCT or the structure. In that case, I mean, most of you are already familiar with those details. But in general, when we talk about OCTs, we know that we can image the front and the back of the eye very nicely. It's a really useful tool for tomography. And OCT has really grown in terms of applications in the last 2 decades. So, over here, I'm just giving a brief comparison of where our system lies with respect to some of the commercial ones. So we have the OptoView, and on the extreme right side, we have the Bioptician, both of which are commercial scanners. And what I would draw your attention to is really the fact that, we can do scanning at a rate which is very variable, and we can scan up to 100 kilos. So we can scan from very slow to very fast. And at the same time, we have probably one of the best resolutions in the market in terms of imaging the ocular tissues. So why are these details so important, and how does it deal with our innovation? We'll go into that. So this is an image of the prototype that we have. It has been converted from a real research prototype to something which can be put in an OPD. This is still at the end of the day a research prototype right now, but we do have a decent compact setup where we have put all the optics in one box here, and then this is the patient interface through which the patient will get imaged. We put a chin rest in front of it. For the photograph, we have removed so that you get a clear view, and then you have the computer. And on the left-hand side, you have this on-face view of in this case, is the cornea. And on the lower right-hand side, you see this beautiful image of the cornea, which has been acquired at very high resolution. Now, something very interesting here is that you would think that this is probably the central cornea, but actually this is an 11 millimeter wide scan of the cornea. And because of the ultra high resolution, we can image the layers of the cornea with immense clarity. So you can look at the epithelium, you can even see the Bowman's layer, and right down below you get a clear view of the endothelium and the descent. And because of the very neat optics that has been designed into it, we can get basically the same ultra high resolution from limbus to limbus, and therefore overcome all the practical challenges which are there with the current OCTs. Because for the anterior segment, we are still restricted by 6 to 8 millimeter of corneal imaging and not beyond that. So in addition to that, we have a very neat setup here, which has been brought to India for clinical use for the first time. So conventional OCTs are basically described in this image here, where you have a light source and your beam splitter. So one portion of the light goes to the eye and the other goes to a reference arm, and through interference you're able to generate the image. But what we have in this setup is also known as polarity sensitive optics. So what we do here is we put polarizers in the path of light, and this blocks certain wavelengths. So when this happens, light goes through the eye in certain phases. Now, because the ocular tissue, it doesn't matter whether it's the cornea or the retina, it has a very lamellar or fibrous structure. These inherent tissue layers change the physical property of light as it passes through. And what we are doing is we are trying to measure these changes in the phases of light so that we can sense disease-specific or treatment-oriented changes in the tissue far earlier than what you would see in, let's say, visual acuity perspective. Okay. So this is an example here which I've given. So I've mentioned here collagen interaction. So if you imagine the cornea which has these lamellar bundles, and we have light passing through it. So because there is a change in phase, we can map out how the collagen fibers are oriented within the cornea. So this slide describes what has been measured ex vivo from human tissue. So from donor corneal buttons, and using X-ray diffraction. This work was done by Keith Meek. He has done a lot of work in this field, and his work is considered the benchmark in this. So if you look at these two schematics on the right, these black and white figures, so for the right eye, you see these fibers that are in the mid to periphery cornea, which is almost oriented circumferentially, but at the center, these fibers are almost oriented at right angles to each other. Similarly, for the left eye, you see the same pattern. However, note a very interesting feature. The right eye is actual mirror symmetry of the left eye. And this is a classical collagen fiber distribution and orientation that exists in the human cornea. But we don't have any means to image or quantify them currently in the clinic. So keep this schematic in your mind, and what we get out from the OCT is this. So if you look at the top two images, which is the retardation images, you see these color distributions, okay, which are basically changes in phase from the PS OCT setup. You have a set of blues here, and as you go to the periphery, you see these transition from blue to yellow through a set of greens, okay. And now if I overlay the schematic which you saw in the previous slide, you can see how beautifully we are able to capture the native collagen distribution and orientation in the human cornea. And this has been mapped exactly in patients. Similarly, for the left eye, this is the figure adjacent to it, you see the same map, but again, this is an exact mirror symmetry. So we have now the capability to look at the super ultrastructure of the tissue in patients, and how do we apply this information? So let's look at the cornea. So we are familiar with keratoconus, steep cornea, very thin cornea. Sometimes they are very asymmetric. And if you look at the collagen distribution, which is in the phase retardation, which is the lower left figure, you see marked reduction in the color distribution. So this is indicating either that the collagen is absent, or the native collagen distribution, which you see in a normal patient, is absent in the keratoconus cornea. So we are able to see that something which you look at topography is also being substantiated by the ultrastructure of the cornea in a patient. Now let's look at a thin cornea here. Now this thin cornea over has very normal curvature. The thickness is low, very similar to the keratoconus which you saw in the previous slide. And look at the color distribution of the polarization sensitive changes. And over here you see that it's very similar to what you would see in a keratoconic cornea. So now we are forced to think that this specific cornea we see here, even though it has a normal curvature, but it has a thin cornea, and because the collagen structures are normal, if this cornea is operated upon, there may be a possibility of ectatic changes, and this has to be, of course, proven as we study this more. But now we have a physical evidence that why thin corneas are at risk for ectatic level changes if they are intervened upon. This is a case of pellucid marginal degeneration, and in this, if you look at the phase retardation map, you hardly see the yellows in the mid to periphery cornea, which shows that the anchoring fibrils are virtually absent. So, all these three cases illustrate how the collagen distribution changes mapped from PSOCT can be used in the clinic. And here I have summarized all of them stacked one over the other. So you have keratoconus in the top, thin cornea in the middle, and pellucid margin in the end. And if you look at the phase retardation maps, which is indicative of how the collagen fiber is distributed and aligned, you see classical differences between each of these states. And keep in mind that all these cornea which I presented here have pretty much the same level of thickness. If you compare this with the normal cornea, so you can see here at the extreme left-hand side, normal cornea is present, and the disease-related changes are very obvious. Okay. If you look at surgically related changes, so we have preoperative cornea. Look at how LASIK, SMILE, and PRK cause patient-specific changes in the collagen fiber distribution in the patient cornea. Now, keep in mind that for all these patients, LASIK, SMILE, and PRK, I've taken cases where they have undergone the same amount of refractive error correction. They have pretty much the same level of preoperative thickness. Yet, each of these surgeries is causing a different level of collagen fiber distribution changes, and this can be a bearing as to how the cornea heals and responds post-operatively. So now we look at retina-related applications. So I want to bring about this case here of geography atrophy. You have the autofluorescence images, which is there in figure A, and then you have the OCT tomograms, which are shown in B and C. And the image D here shows the image that is generated out of mapping the polarization changes in the phase of light. And clearly you can see in this orange marked zone that there is accumulation of drusen. So this is fine and good. Now let's look at a case of Stargardt's. Again, look at A. This is an autofluorescence image. And let me just take your attention back to the previous slide A over there. The autofluorescence images look pretty much the same, yet they are two different diseases. And if you look at the polarization changes that are being mapped through the OCT, you see over here in image D, how a different phase change is being captured by polarization in the Stargardt's case versus, I'll go back to the previous slide, in the geography atrophy case where it is more in the oranges and reds. So this is a clear indication that the accumulation that you see in the tissue has different biochemical and biophysical interactions in these two diseases. And now you have an imaging readout out of this scanner, which can be used as these patients are treated further. There are additional applications. For example, this is known as DOPU. So in this case, it's DOPU is a form of analyzing the polarization signals. And in this example, what has been shown is that the signal itself can be used for segmentation of the tissue layers, which becomes very challenging in case of disease cases, because normally when you have a disease, you look for intensity changes. Most methods will look for intensity changes through the thickness of the tissue, and they will use that to segment out the layers. But here, the change in the polarization state of the light is used as a marker for segmenting the different layers of the tissue. So we are not too worried about where the pixels are too bright or less intensity. The biophysical property of the tissue is allowing us to segment. So if you look at figure D, there is a faint red line, which is allowing us in segmentation of the layer. This is another example of visualization and quantification in neovascular AMD, where in figure E, the arrows point where specific problems are there in the tissue, and this is optically determined. We don't need to do any further image processing after this. So, so we have started in our clinic primarily with retinal degenerations first, and on the left-hand side, you have the OCT tomograms. On the right side, you have the phase retardation map, which is the direct output from PS. And if you look at a normal retina versus Stargardt's, retinitis pigmentosa on atypical RPE, you see distinct changes in the way the light is modulated as it passes through the ISOS layer. Now, keep in mind that in all the cases that I have picked up here, Stargardt's, retinitis pigmentosa, atypical RPE, they still have very good vision. They are all in the 6.9 range. Yet we can see already severe disease-related changes in these patients, and it has not yet manifested at the visual acuity level. This is how a normal eye, this is how we are generating readouts from our OCTs currently. We have our beautiful OCT scans, which you're all very familiar with. We can also generate a fundus map through the OCT on fast reconstruction, and then we're also getting for different layers. So you have the top row, which is ILM, the middle row ISOS, and the last is RP. And for each of these layers, as you can see, we are generating PS maps. And this is where the ultra high resolution really comes handy, because some of the retinal layers are 10 to 20 microns thick, but while some of them are in hundreds. So therefore, higher axial resolution is very important. And this is some additional processing which we can also do, where we can remove the cornea PA polarization sensitive changes that appear on the retina, and therefore give only retina-specific ocular changes to the PS signals. And we are working further towards building analytics so that it can be used for a number-based quantification by the clinics. So to summarize, we now have patient-specific visualization quantification for birefringent structures. We are looking at treatment-specific response in the cornea and also disease-specific modulation. And the ultra-high resolution and wide field imaging is really helping us to screen patients for retinal therapies, particularly the regenerative medicine, which Orko spoke about in the last presentation. And that's where we are seeing applications building up through the scanner. So where does the future? I will end with this slide. So we have been doing tomography for decades now, as you can see from quite a few talks that have also been done so far. So we have tomography to assess thickness and volume. Through this presentation, I have talked about birefringent properties. So the fibrous, the changes induced in the polarization state of the light that is being induced by the fibrous or the collagen network of the ocular tissues. And then we also have the advantage of varying the speed. Why varying the speed is essential. This is some new things that we are working on. Hopefully by the next presentation, we'll have some data to show that if we alter the speed, if we can image very slow, we can also image the cells and the nerves in the tissue without the requirement of adaptive optics coming into play. Okay. And eventually all of this has to be combined with robust AI and deep learning to facilitate the clinicians, because these are three different sets of information, and it's going to be quite challenging for a clinician to interpret all of that. And why AI and deep learning? So we are in the COVID area. So I'm going to talk about briefly in few lines that we are all familiar with these curves now. So these are not COVID cases, but what this curve is essentially showing is that we have onset of a disease which is marked by an increase in the curve, and then you do a treatment which can be drug or a surgery, and then it's supposed to normalize. Now with each of these features here, which is tomography, birefringent, and response in the cells, it's quite reasonable to expect that the response of these structures will all be different at different stages of disease. And this is indicated by the different peaks at different time points. But if we were to use all these temporal changes that are being mapped with the ultra-high resolution scanners, we need to come up with robust AIs to combine all of this information into one robust tool. And this is where we are moving towards in building and developing these unique tools for use in the clinics in the near future. So with this, I would like to conclude. I'd like to thank Netherlands. It's been really great journey with them for all these years. Dr. Shetty and the Indian clinicians, my team, and my collaborators also, Dr. Christoph and Michael, were very instrumental in helping us with this super resolution imaging. And also, not the least, the funding from government and several non-government agencies. Thanks.
Thank you so much, Abhijit. That was very comprehensive and very exciting journey that you've had. We'll probably keep the questions for the end, inviting the next speaker, Dr. Swaminathan Setu. He's a scientist at the Aurolab, and the next three talks are the rapid fire talks. So they're just going to take about 4 or 5 minutes each, and then we're going to wrap up this session. Dr. Swami, over to you.
Right. Good evening, everyone. I just need to share the screen, but I'm not being allowed.
Abhijit, you have to stop sharing your screen.
Yeah, I'm doing that.
So while that's happening, I'm going to talk about the molecular story, a series of molecular series of molecular short stories, I would rather. It's going to be a patient-centric approach, and it's about how to use molecules for prediction, prevention, and precision therapeutics. Okay, so this is the cast, the molecular story cast. The clinical sciences and the basic sciences have come together to answer one question: how to predict patients before they even get any kind of conditions. Whatever you're seeing in imaging is something which has already happened. How can we catch them early? The second question the clinical team was asking was, can we have a unique prescriptions for different patients? That is precision medicine. And the only thing will come about of this whole thing is by understanding the molecular aspects. And this is the entire technology of the molecular story. We have an array, as Abhijit and Arko pointed out, thanks to the hospital and the various funding agencies for providing us these various technologies to dissect the molecular story. And you guys are the Superman who stops the disease dead on its tracks and save the patient away from progression. And this Superman, that is you guys, the clinicians, will only be able to do this if you have the right information. Information is power. And every successful clinical outcome is based on the wisdom or knowledge of the clinician, particularly in modulating which molecules, which is disease causing or which is disease resolving. And molecules is at the very foundation of the entire pyramid. Molecules instruct the cells to respond, and the cells respond, the tissue responds, the organ responds, and therefore you'll have the disease phenotype, be it signs or symptoms or structural changes like imaging or biomechanics, whatever the case is, the preceding event is at a molecular level, and it could be thousands or millions of interactions which is contributing to it. So we have to know it to better assess it and rein it in. And from the etiology to the phenotype, it's not like an IBCM going from point to point. It goes through a variety of factors. And knowing these molecular pit stops can give us new directions in terms of therapeutics. This might look very similar. This is how the biomarker discovery looks like to somebody who's not familiar. It's, yeah, they're all planes lying off. But if you put a grid, and if you know the altitude, direction, and velocity and size, you know each one of them have a unique destination. This is exactly how the biomarker discovery at NN is happening. And I'm going to give you 5 stories, 5 very, very short stories. Dry eye disease has been a major problem for a whole lot of population, and we don't have a lot of medications. And most of the common ones are cyclosporin, tacrolimus, and a bit of Zedra, which is not yet in India. But if you notice, each one of them is targeting a particular molecular aspect, which propagates the disease. Yet, not all patients respond. And that's what we asked. Who are these patients and what do their molecular pattern is so that we can have a very targeted therapy? And we did come about with this. All these factors which you would have seen in the literature as well as in our cohort in NN, these are the various factors which have contributed to the disease. But each of these subsets have been very high or low in different types of patients. So if you have a molecular signature for each and every patient, you have a unique set of therapeutic agents who they will respond to or which they will respond to. This is called bespoke treatment, and the molecular story of each of these patients driving the disease becomes center stage. We talked about the keratoconus phenotype. The same question applied. The clinicians were asking this question: who will proceed? Yes, the imaging will show whether they have proceeded or not, but we wanted to know who is proceeding. Once again, we have a set of molecular markers which clearly indicated a certain set of progressors have these molecular patterns, and we already have the drugs in place so that we can prevent the progression from happening. The story number 3, haze, post-refractive surgery haze. And one of the best work done by NN and Aurolab team was predicting a clear indication who will develop haze. And how did we find out? Everybody knows TGF-beta drive stage, but as Dr. Javed is saying, sometimes we have to redefine anatomy, and likewise, we have to redefine the molecular biology and cell biology. And the work done by Arko and Dr. Roy Shetty clearly showed that it is not the TGF-beta, it is PREX1, which predisposes these patients from developing fibrosis. It basically becomes an amplification factor. And hence you have different therapy. And this is with Dr. Padma Malini and Dr. Namita Dave, and they clearly asked why usually sarcoid-associated uveitis is a case of exclusion. Because you don't fit all these TB, viral, and autoimmune, then it could possibly be ocular sarcoidosis. But we did find a molecular evidence that this increased CD4-CD8 ratio in the eye was very, very predictive of ocular sarcoidosis. Story number 5, once again, it's about retinopathy of prematurity driven by Dr. Anand Danekar here. So the question was, when you have the baby visiting, a preterm baby visiting you, there is no ROP, but when they come back for a follow-up, they do have ROP. Dr. Anand's question was, how can we predict it? Yeah, we are imaging, but how can we predict it? We looked back at the molecular factors and we collected from the tears, and we clearly found out that the VEGF and another angiogenic factor, angiogenin, when you did a particular bit of mathematics to it, along with birth weight and gestation age, we could clearly predict who is going to develop ROP, and it's going to be very useful at a very good odds ratio with good amount of specificity. Of course, it's a small cohort of 30 patients. Okay, so molecular aspects comes very handy in these cases. But one is not enough, is what we have learned in NN. We are moving away from single biomarker to multi-marker strategy, and we are developing molecular diagnostic tool, and we want to integrate it this way. integrate molecular with biomechanics to imaging. So the IBM stand is what we are going to take. We integrate clinical information with all these to bring about absolute prediction, precision in therapeutics. And molecular diagnostics is going to revolutionize clinical ophthalmology. It is already on, and I think we should be part of it. Thank you.
Thank you, Swami. We'll go over to our next speaker. That would be Dr. Thirumilesh, good friend from my department. Having completed his training at RP Center, he's been a consultant with us, and he's also consultant and SME for SIT to Play Private Limited, Bangalore, for AI solutions in retina. Over to you, Thirumilesh.
Okay, thank you, Dr. Chaitra, Dr. Anand, and Entod for giving me this opportunity to present the slides. Are my slides visible?
Not yet, not yet.
Okay, it should be on now.
Yes.
So basically, the topic that was given to me was new toys and tools. So what I would be presenting is how did the validation part of it, they're not moving. Okay.
Yeah,
so how do we validate it? So as already stated, that is my financial disclosure. I have been a consultant. So why we went into doing the AI part to the clinical application was basically for this reason, that diabetic retinopathy is a huge problem in India, and majority of the patients present with very advanced stage of the disease. So we wanted to detect them early by going into the community and catching them early. Thanks to Dr. Kim, majority of these slides have already been like kind of repeats. So what we do are we take fundus image, we ingest it into the cloud platform and see whether the classification and the model does, and then we put it onto the reporting engine, and then that's how the output is generated. So this is basically an annotation tool. So if you are planning to innovate based on what you could add onto the fundus, is that you would have to take an image and identify each and every single pathology and annotate, and that's how would help in training the model. So the output of our particular collaborators model would look like this. It would give you an outcome where it would show whether you have diabetic retinopathy and in what zones it is affected, and what is the predictability rate of it being either a proliferative or a non-proliferative diabetic retinopathy. The results of which are pretty encouraging. The referable DR was agreeable between the annotators and the model in up to 95% of cases. However, when it came to non-referable DR, it was about 90%. I'll show you why this happened. Is because when it comes to the mild DR cases, the overall sensitivity with which the machine picks up was only 41%. Now, why AI for OCT was because it helps in reducing human error at reporting. It can be trained for identification of biomarkers, which would be the final goal. However, the Drishti platform, where we have trained the AI system to do an automatic analysis of OCT, is at a level where it can identify pathology. So the study design was, we took OCT scans from patients during the period from 2016 to 17, and annotations was done by ophthalmologists, and then it was compared with the machine learning protocol. So we followed this particular classification, which was by Kim et al. From American Journal of Ophthalmology, published in 2006, where the pathology was identified as cystoid, subretinal fluid, tractional, or a combination patterns. So this was the AI annotation tool, where we gave the AI a certain set of images, and we saw whether it would kind of identify the images or not. So the precision was about 98% with the sensitivity of 91. And the FN score in general for both identification of normal was about 95%, and for identification of abnormal pathologies was about 95%. Now, I would also like to show a third study, which was for identification of refractive errors. What we did was, there was collaboration with a Korean company who came to us with this particular application of AI, where there is a software which you could download to your Android or an Apple iPhone, and that particular phone, without using any other instrument, would be able to give us a refractive error. So this is how the app, once downloaded, looks like. So all you have to do is align it with the eye of the patient. In this particular thing, we had also included kids between 1 to 5 years of age. So at 1 meter, using the app which is downloaded onto an iPhone, you can take a picture by aligning the eyes within the slots which are mentioned, and then that would give us what is the kind of refractive error that this particular patient would have. So the range which the company claimed was that it would give accurate results between -5 to +5, and the cylinder should be less than -3 diopters. So once we did the study and validated, we found that it was only true up to 70%, and the agreement with AR and manifest refraction was about 70, but it had a good agreement with automated refraction and within the specified range. However, there were up to variations up to 0.75 diopters for sphere in more than 30% of cases. I'd like to bring attention of the panel and also the viewers to this particular slide, is that although Google's AI was super accurate in the lab, but when they took it to the real life situation, it was slightly different. However, what the article also says is that if the person who is taking the images is well trained, then it can really take us to the next level. So whether AI or not, how well we will be able to take it to an outreach also depends upon who is operating it and how they are operating it. So validation is really, really important to really take AI into the field, and that's the way to go.
Thank you so much, Dr. Tirumalesh, for briefly yet eloquently touching on this new AI and the exciting possibilities. We'll move on to our next speaker, Dr. Gagan Dudeja, who's a consultant orbit and oculoplasty, and he'll briefly tell us about the innovations in retinoblastoma.
Good evening, everyone. I'm standing between you all and your dinner probably, so I'll try to wrap this up fast. So what are our innovations in retinoblastoma? Just to briefly take you, we classify retinoblastoma from group A to E, starting from small tumor to big tumor. And the problem with us is the developed world sees the smaller tumors. We get all the bad tumors. Once tumors are that way, our prognosis is not good, in spite of all the treatment. So our problem with retinoblastoma is the eye salvage rate, and in addition to the chemo complications. So no matter what chemo you give, there are still going to be some complications. So how can we address this problem? So in order to improve outcome, these are the areas where we had a question that can we have a better and a selected targeted chemotherapy or any other therapy? How can we increase the effect of existing chemotherapy? How can we reduce the doses of existing chemotherapy medications so that the adverse effects are reduced? We also are looking at the biomarkers which will help in diagnosis, prognosis, and follow-up, and also genetics of retinoblastoma. Part of this and how we go about it was covered by Dr. Arko. I'll just cover the first three things and share our experience with it. So for first part, we looked at a cationic antimicrobial peptide, CAP or CAMP as it is called, to see whether it can work against retinoblastoma. It's a naturally occurring peptide, small chain, works against bacteria, virus, cancer cells, and multiple things. So it has got a positive charge. So it will get attracted to the negative charge of the cell wall of bacteria as well as retinoblastoma. So retinoblastoma cell wall has a negative. So can we use this against that? So simple thing is the first experiment is to put the peptide on the retinoblastoma cell line, very one cell line. And if you see here, these are various peptides. The round one, HC3 and HC5, are showing killing of the retinoblastoma cells. So they are acting against. Now to see whether they're active or not, the next step is to put the same thing on a retinal pigment epithelium cell line. And we can very well see that these peptides are not killing the retinal pigment epithelial cells. That means these are selective towards retinoblastoma cells. And then we use the same thing along with topotecan to see the effect on retinoblastoma spheroid model. Tumor spheroids mimic the retinoblastoma tumors. With topotecan, if you see, all these spheres get disrupted. It's like a nuclear bomb. Everything gets disrupted. Whereas HC3 and HC5 demonstrate more selective and controlled killing of retinoblastoma cells. So this is kind of a soft killing. So they are not going to cause that much collateral damage. Also to see whether we can have effect with existing chemotherapy medications. As I told, we can reduce the dose. We checked for synergism along with topotecan in a checkerboard assay. So here if you see, the FICI with topotecan is 50. So with this, we know that it can work towards a synergism with topotecan. That's a clinical significant that we can use this drug along with topotecan. Now the next step was what is the mechanism of killing by these caps. Normally, they are supposed to be acting on the cell wall, causing damage to the cell wall because of the positive charge which gets attracted to the negatively charged cell wall. But it has got a different mechanism with different organisms. So the mechanism with the bacteria is different than with the cancer cell. So how were they acting upon cancer cells? For this, we went ahead and did the experiment. And if you see here, this is a DNA damage. This is with topotecan, and this green one is a DNA damage, that phosphoryl H2AX. So the DNA damage with topotecan is quite significant and quite massive. Whereas if you look at the DNA damage with the caps, it's much more controlled. There is a less DNA damage, but more cell death with caps. So it is a much more controlled thing. And it also proves that it is also targeting mitochondria. So to prove that is our next set of experiment. Here, if you can just see, this green one is a cap cell which are labeled and they are sticking onto the mitochondria. And this is the mitochondrial outer membrane state with VDAC. And on the merge, we can see the cap cells are along with the mitochondrial cell wall. So this is a clear proof that in retinoblastoma cell killing, the mechanism of action involves mitochondrial damage and release of cytochrome and consequent damage to the cell. And what else can be the pathway? So how does the final apoptosis happen? So we looked for that. And here, if you see, this is a caspase pathway. So you have a initiator caspase and a executioner caspase, and both these pathways are activated. So this is a proof of the caspase pathway activation. So we have these 3 steps of mechanism of the action of caps against retinoblastoma cell, which is previously not reported. So what we found with this study was that with caps, we could have a novel molecule which can work against retinoblastoma cell as a selective targeted therapy. It can also work synergistically with topotecan, and it can also, the mechanism of action was described by us for the first time. The second half of another selective approach towards was to look for EMT. Epithelial to mesenchymal transition is known in tumor cells. It gives us, EMT helps in tumor initiation, invasion, metastasis, and most importantly, cause resistance to chemotherapy. So we also know the epithelial markers, which are onto your left, and onto your right are the mesenchymal markers. So we tried to look for this pathway, whether this pathway is working in retinoblastoma cells or retinoblastoma tumor, and if it is working, how can we confirm that? So here we took the tumor's tissue and we checked for the differential expression of EMT markers. So these all markers were found to be upregulated and these were downregulated. So we went for the further step and did the same markers in a retinoblastoma cell line using qPCR. And if you just focus over here, you can see that NCAD and vimentin is high, which is indicating of an advanced stage of tumor. There's a high FN1 here, SMA, twist, which are associated with tumor metastasis. And high BCL, XL, and AKT are associated with resistance to the tumor. So the markers of EMT are there in retinoblastoma cell line. So for protein expression, again, the same experiment repeated with Western blot. If you see here, we have vimentin and twist here, which indicates that it's reduced, and E-cadherin is increased, which again indicates activation of VMT pathway. And alpha beta catenin and beta catenin again, if you see, are activated, which indicate the Wnt signaling pathway. So we have a role of Wnt signaling pathway based on these two. So the important thing here is for EMT, because it is already there in multiple mechanisms, there are multiple medications already available. So you can see a long list of medications, and there's a longer list available which work on EMT pathway. So once we know the drugs, and we know that RB1 dependent retinoblastoma gene has an EMT pathway, we can use these medications to provide us new therapeutic intervention. So it will involve repurposing of these current drugs. And the most important thing here we could find was Wnt signaling pathway driving EMT markers in retinoblastoma, which is a novel finding. Thank you. Sorry, I went too fast because of lack of time. Anything is there, you can ask me the questions.
Yes, I think, but you covered it well, Gagan. Thank you so much. It's just that, there's so much to put in these talks, and it's always not easy to explain. So we do tend to, take some more time than expected. Our last speaker for the day would be Mr. Nikhil Masurkar. He's the Executive Director and the Research Head of Entod Pharma. And he has, got a whole lot of degrees under him. And I think he's well equipped to talk to us about, how to go forward with innovation and research. Over to you, Nikhil.
Thanks, Dr. Chaitra. Firstly, before I start, I would like to thank Narayan Nitralaya, Dr. Rohit Shetty, for giving me the chance to be part of this webinar and speaking. So my talk today is about innovation, quite a broad topic. And I have the privilege also of being the last speaker of this webinar. So, but I promise that I'll take as less time as possible. Firstly, I would like to declare my financial interest in the company that I run, Entod Pharmaceuticals, and the Entod International Group. So for me, the innovation is, simply finding a new solution to an existing problem, and it's always been like that for me. Now, the whole world is going through a major crisis right now, and COVID-19 is on everyone's mind, and it's affecting everyone in the largest possible way. So the question is, what is pharma in general doing to solve this problem? Now, I'm not going to go through this in much detail, but just to give you an overview, because I don't want to overburden you on information on COVID. But right now we're developing new treatments. So we have the antivirals, the hydroxychloroquine, remdesivir, favipiravir. But most of these have just found to reduce the recovery time from say 15 days to say 11 days, which helps you can get out of hospital a bit faster. But that's all they can do at this point of time. The other, potential treatments are the antivirals with the immunomodulatory effect. These are molecules like baricitinib, which can reduce the levels of cytokines that cause damage to lung tissues. They can be used in moderate to severe cases, but right now it's just being tested, on existing patients who have COVID-19. And then obviously you have convalescent plasma transfer, which has been done for quite a while, but it's currently trying to be tested in critical cases. None of these approaches are new; they're just being currently tested, and it's the only time will tell how effective they are. But what we're really waiting for is the big one, the vaccine. The good news is there's about 120 vaccine projects around the world right now. 5 of them approved for clinical trial. The good news for India is that the vaccine that is currently being developed in Oxford is showing a lot of promise, and that will be manufactured in India very soon. I won't go into details of that. But one thing we know for sure is that we are unarmed right now, and we will be for at least a year. So during this period, the pharma industry could start a whole new wave of innovation. So what can the ophthalmic pharma do? Well, I think we can transform. I think we've been given a new platform to really make these changes now. And one of the ways we can do it is by collaborating with technology firms and doing something very different. Like, for example, building a mobile app to achieve social distancing in ophthalmic OPDs. You've already probably seen this in the news. You have the Arogya Setu, the Singapore Trace apps. A lot of these apps basically use Bluetooth, low energy Bluetooth BLE beacon technology. That's already there in most of your phones. So the way it works is that you just download a simple app. 2 people, when they come close to each other, less than 1 meter, it will kind of beep. It will alarm them so that they can keep their personal distance. What we can do is, what Entod is already looking at, is how to customize it for OPDs. Imagine a doctor sitting in his cabin knowing that social distancing is being practiced in his waiting area. That would be great. The other thing we can do is we can design innovative telemedicine apps customized for ophthalmology. Now, there's already a lot of these there, and recently the Ministry of Health has also issued practice guidelines on telemedicine. This is going to be a big thing moving forward as well. So it's kind of legal to do it as well now. There's a lot of these apps around. What we can do is we can customize these. We can work with these companies, customize it towards ophthalmology. Or we can launch innovative products to help reduce patient waiting time. So we already have an intraoperative essential to eliminate the need for pre-op dilation altogether. We're going to be launching a product called Phenocane Plus. So this can reduce patient waiting times and also surgery times. So in a way, it would be a COVID essential. Or we can transform online ophthalmic continuing medical education in ways that it's never been done before. So we're going to be starting an ophthalmic experience academy. Now, I know we're doing a lot of webinars and people are asking why so many. What we want to do is we don't just want to do the webinars. We want to convert these webinars into learn-on-demand web pages. So we want to then edit the video sections based on specialty, have breaking news articles, hot topic articles, debate sections, online quiz, question and answers, and then basically make it a good educational tool. So the post-webinar activity for us would be more important than just kind of streaming it live and people tuning into it. So what we see is a transformed pharma that will innovate more than just in drugs and vaccines. What I see is a transformed pharma that will be more of a service where the product will be just one part. So by that, I would like to thank all of you for being part of this webinar. And if there's any questions, I'm sure the panel can ask me. Thank you very much.
Thank you so much, Mr. Nikhil, for telling us how in different ways pharma can engage, and specifically Entod. And I think taking us through the layers of engagement and the future that you foresee, not just in this difficult time, but to moving forward. I'm just going to summarize very quickly what we think we have learned, putting together what all the speakers have had to say and the panelists. And then if there are any more words from any of the panelists or speakers, we'll take that before we close for this evening. What we learned, I think, is first figure out the community need. And for that, there is a felt need. As ophthalmologists, we are dealing with subspecialties, and you figure out what it is in your subspecialty that might be missing. Then, of course, as many pointed out, you have to have that curious instinct. Keep that curiosity alive and put in all your expertise, keeping that alive to reach that solution. Once the idea is generated, we would need mentors. These mentors could be people, could be an institute, could be organizations, could even be the government, and of course, funding agencies. We've learned that AIOS can be one of those partners, Department of Biotechnology could be, industrial partners, some of them who are speakers, and even international collaborators. And then passionately go on with that one idea of yours till it is, you go over several iterations till finally it's ready. Once it's sort of ready, you have an option to either publish, to put in a patent, or to productize. Ideally, we would want all three. They're not necessarily in that order, but it would depend on the question of whether to patent or not, publish or not. Dr. Suven has very nicely summarized it. To patent would be how good is that idea for it to become a financial viable product. So for that, the three pillars to decide are: one, the novelty of that idea; second is the inventive step; and the third is what is the industry applicability. Once you've answered these three, then you are at that crossroad where you can either reinvent or go on to productize that, as many of our speakers and panelists have done. And eventually, once your product is made, it should reach where it's most needed. And that is at the outreach where maximum number of our countrymen and the world can benefit at an affordable cost. Never use the word cheap, never use the word even low cost, definitely use the word affordable. And I think that's what we've really learned today. I really thank all, of course, the speakers and the panelists, but over 7,500 people who have connected through 3 of our platforms. We kept getting the statistic through all the 3 social media platforms. That's a very fantastic number for a Sunday evening. Thank you so much for being part of it, the speakers, the panelists, my co-moderator, Dr. Chaitra, and Entod Pharmacy for actually having the foresight to support a webinar of this scale and this theme. Before I sign off, any of the panelists and speakers have anything to say, we'll do that and then wish all our viewers a good night. Yes, sir.
Can I? Yeah, it's a fantabulous things that you've put together. Congratulations to the Naranitla team as well as Entod. I would like to say that the development of a product or innovativeness or lateral thinking has been there for time immemorial. So when I started my practice in 1980 in India, it was never difficult to implement whatever I wanted to do it. So I'm a clinician basically. If I wanted some tool, some equipment, I'll just call my friend. He'll develop it and bring it back to it. Maybe working, may not be working, but there are roads of rectifying it. That's been continuously on. Even recently, Aurolab has developed a lot of instruments to me. And we developed a lens which has on a hydrophobic platform. It's never done in the world. They made a lens of cylindrical power of 14 diopters. That's amazing. They took about a month or 2, but that's the power we have in India, that you can think something and you can implement it. So I had a lot of surgical instruments made by Aurolab, Epsilon and various other people. They're very friendly people. Even the pharma side, you think of something, they are there to help you out. I think at India, where the regulations are not very strict as of now, if you have no interest in either, so I have no interest in publishing anything that I do. So for a clinician like me, this is the best place in the world where I have thoughts, where I have a patient who will get benefited, where there is a friendly industry who can help us out and we can implement and see the results. So this is what I would like to put across all the people who are here. Thank you.
Thank you so much, sir. Yes, Dr. Sabin.
Yeah, so what I wanted to say was we have to change our mindsets. The younger people are doing. Yes, we are getting more into innovation, but it has to be more organized. If you look at the West, they are very much into entrepreneurship. So that is what we are here into instant glorification. We have a device or an invention, we come up with it, we publish a few videos, a quick article in a non-peer-reviewed journal, and we are very happy with it. And then somebody steals our work and does it elsewhere, and then we are left looking like fools. So we have to have that more organized, and our innovators need to be educated, and there's a lot of hand-holding which has to be done, which has to come from in a very organized manner.
I fully agree.
Well, if there's nothing else to be said, thank you once again. And thank you again, all the viewers, analysts, speakers, and thank you, Entod.